Welcome back to the second day of International Conference on Radiation Research, Impact on Human Health and Environment, and fourth biennial meeting of Society for Radiation Research. We hope you had a productive and insightful first day. We have an exciting lineup of speakers who will share their expertise today. And for the first session, I would like to invite Dr. Satoshi Tashiro, Hiroshima University, Japan, and Dr. Wahab Ali, Scientist F and Senior Deputy Director, ICMR Patna, to chair the session.
Okay, so now today is the second day of this meeting, and I'm very happy to start the first morning session now. So I'm Satoshi Tashiro from Hiroshima University, and I will co-chair with
Dr. Wahab Ali from ICMR Institute, Aiims, Patna.
Thank you very much. So then we have one announcement. The speaker will be changed from Nils Cordes to Soji Sebastian. So this is decided just now because of some administrative reason. So Soji, probably there is some slide. Can you start a slide? Probably they have there is a, okay, so now we will ask Soji Sebastian to give a talk. So he is from Canada, and he has established very fancy machine and then he analyzed the epigenetic change, and he will give a presentation about the epigenetic basis of radiation health effects using the muscle tissue. So Soji, can you start?
Am I audible? Okay. Good morning, everyone. And first of all, I would like to thank the organizers for giving this amazing opportunity to come here and present my work. Title of my talk is Epigenetic Basis of Radiation Health Effects, Tissue Regenerative Capacities Modulated by Ionizing Radiation Exposure, ionizing gamma radiation exposure. I'll give a brief overview, biological effects of LDR on human stem cells, human muscle stem cells, for that matter, an omics-based approach. So we use gamma ionizing radiation from a cobalt-60 source, and we expose stem cells from biopsies, biopsy-derived stem cell cultures, exposed to the source for 10 and 100 milligray radiation exposure. Then we look at biological effects at time 0, time 6 hours post-radiation, and 14 days post-radiation exposure. And the kind of endpoint analysis, as you can see, metabolomics, followed by epigenomics analysis by ChIP-seq, then some transcription profiling by RNA-seq, then some functional analysis by using these cells, differentiate them into muscle tissue called myogenesis, and look at the pathways which is modulated by ionizing radiation exposure. Okay. Just to give a brief introduction about the potential effects of biological effects, or potential biological effects of radiation exposure. As you know, high dose exposure causes chromosomal damage, something above 2000 milligray, or even at 500 milligray above, you started observing radiation sickness. But in cell cultures, I think you see chromosomal damage upon exposure to high dose of radiation, high dose of ionizing radiation, which is accompanied by gamma H2AX foci appearance in these nuclei exposed to radiation. But in the case of low dose ionizing radiation, I mean, you don't really see a lot of DNA damage, but at the same time, we expect some stimulatory effect from low dose exposure. That's what I'm going to actually discuss, and what are the potential benefits or potential repercussions of this exposure. And we apply this particular fundamental in the context of skeletal muscle regeneration, okay, skeletal muscle regeneration. So if you look at skeletal muscle, an uninjured muscle is, the individual fibers are hexagonal in shape, all the nuclei kind of arranged just around the periphery. Upon muscle damage, you see a tremendous amount of inflammation, which you can see a lot of immune cells are actually migrated to the site of injury, and within 21 days, the entire area is being regenerated and relaid with the newly formed muscle fibers. And just because of the presence of muscle stem cell in these tissue is making this muscle tissue, to regenerate the injured area because of the presence of muscle stem cells. And we are looking at the effects of ionizing radiation on these precious, rare muscle stem cells. So first thing what I did was, we performed metabolomic profiling of muscle stem cells exposed to 10 and 100 single acute radiation exposure, okay? Single acute radiation exposure from cobalt-60 source. And immediately we saw at 6 hours post-radiation exposure, we see tremendous buildup of lactate in these cells, lactate, okay? With a concomitant decrease in pyruvate, that is suggesting that there could be a potential metabolic switch from oxidative phosphorylation to glycolysis, okay? This is an indication of a metabolic switch. Then what we did was, we looked at some of the prior art, published literature, that suggests lactate is actually acting as a major epigenetic carbon source, where lactate can modify histones in the nucleus of the cell in the exposed groups, okay? And what you can actually interpret in my situation, where low-dose ionizing radiation enhances the production of lactate, and that lactate can actually modify histones in the nuclei of the exposed cell, okay? And we tested by immunofluorescence analysis. As you can see, 10 and 100 milligray kind of showed more staining in histone acetylation, Then we performed mass spectrometry profiling in collaboration with Active Motif in Company USA in Carlsbad. We looked at close to 120 different histone modifications in these cells, exposed cells. And irrespective of the doses, you see dose-dependent and dose-independent responses we could see in the detection of histone modifications, okay? As you can see in 100 milligray exposure scenario, you seems to have a lot of substantial increase in histone modifications, specifically acetylation, which is inconsistent with the prior art or prior literature. And on the y-axis, or the color notations are actually, when you detect more of this histone modification, it's in red, otherwise it's in blue. So that kind of contrast I could see in my analysis. Then we looked at overall histones, and the amount of acetylation were quantified by mass spectrometry. And as you can see, both H3 and H4 canonical histones, those have more acetylation was recorded in the exposed cells 6 hours post-radiation. And more importantly, this is after 6 hours post-exposure, but even after 14 days, we see changes in the exposed cells. In particular, the canonical H3K27 acetylation and H3.3K27 acetylation. Both of them have had more of histone acetylation compared to the old or aged cells post-exposure, okay? So we could see that particular change. So I can explain a little bit more in here, the experimental setup, where you expose cells to low-dose ionizing radiation, you age them in culture for 14 days, and you assess the histone levels by mass spectrometry. So you have a young control and an old control or aged control. So compared to the aged control, you have in the exposed groups, you have more acetylation on both canonical and the memory, epigenetic memory marker H3.3 as well. So that is very outstanding, and I see that as a potential results for future diversification of the project. Then we performed genome-wide ChIP-seq analysis by cut and tag, and we could see in young cells you have a robust amount of histone acetylation at H3K27 was recorded, but upon aging, you lost them, or there was a consistent loss, and that loss was kind of mitigated up to some extent in both 10 and 100 milligray single acute exposures. And the loci was regeneration for loci, which is implicated in insulin signaling, okay? It is implicated in insulin signaling as per the prior art. Then I looked at one more loci where I see a loss of histone acetylation in the aged cell, but in the 10 and 100 milligray exposed cell, we see an increase over and above the control. So which is indicating that basically low-dose ionizing radiation exposure enhances acetylation on H3K27 histone enhancer mark, okay? And again, caveolin 1 and 2, which is the gene loci I looked at, is again implicated in insulin signaling. Then I looked at what is the outcome, what is the ramification of this particular treatment. And we see that when I basically differentiate these cells into muscle fibers, we see more muscle fibers being produced in the 10 and 100 milligray radiation exposed groups. Okay? As you can see here, nicely formed muscle fibers in green in color, which is basically quantified by immunofluorescence microscopy. And in 10 and 100 milligray did have a pronounced effect in terms of myogenesis. Then we tested this in vivo, where young animals were exposed to 10 and 100 milligray. Then I induced an experimental injury in their tibialis anterior muscle and looked at the efficiency of regeneration post 21 days post-exposure. And this is what I did. I mean, my initial observation was, totally, there weren't any gross damage or defect in the exposed individuals or exposed animal groups. Rather, what we see was actually the 10 and 100 milligray exposed group had muscle fibers in the regenerated area which had a much higher fiber caliber or much bigger size, which is an indicative of a better regeneration in the exposed group. And then we performed some transcription profiling. We see that both 10 and 100 milligray exposed human stem cells possess or showed genes involved in insulin signaling pathway. For example, IGF-1, insulin growth factor 1, or insulin growth factor binding proteins. And most of them have a potential effect in myogenesis, a pro-myogenic effect. So both in ChIP-seq analysis, right from metabolomics to ChIP-seq to transcription profiling, has indication suggesting that low-dose ionizing radiation somehow enhances insulin signaling, okay? So that's my take. And those genes involved in these activities were kind of belong to receptor tyrosine kinase function, RTK function. Then we tested the scenario in our gene ontology analysis. Again, those terminologies were showed up, tyrosine modification and tyrosine phosphorylation, which is again implicated in insulin signaling. To begin with, the first quarter, the upper quarter, where when you actually treat cells with IGF-1, one of the candidates in our gene expression analysis, we see an improved myogenic differentiation. And when you treat cells with insulin, also you see an enhanced myogenesis. And the candidate from my ChIP-seq analysis, both 10 and 100 milligray, showed an enhanced acetylation on the REC4 promoters and enhancers. When I treat cells with REC4 or regeneration 4 protein, you see an enhanced myogenesis as well. And same thing, I observed from our metabolomic profiling, the candidate lactate, what we found in the 10 and 100 milligray exposed scenarios, that lactate also, when you added in the culture of muscle stem cells, it could enhance differentiation in a bigger way, substantiating the fact that low-dose ionizing radiation and associated modulation of the gene expression is actually building the vibe towards a better myogenesis. And I took this information and applied in biopsy-derived cells from healthy type 1 and type 2 muscle biopsies or muscle stem cells. So to begin with, normal biopsy-derived muscle cells, you age them in culture, exposed to radiation and age them in culture, you see a tremendous increase in myogenesis. And if you treat it with insulin, along with the irradiation scenarios, you see an enhanced effect. But that effect was kind of disappeared when cells from type 1 individual were exposed to low-dose ionizing radiation and insulin treatment, did not show that effect at all. But rather in type 2 derived muscle stem cells, we see a greater synergistic effect. And this is my observation, and my interpretation is low-dose ionizing radiation has an effect which is being modulated further or amplified by treatment with insulin, only in type 2 muscle stem cells. Then we are focusing more on the signaling pathways and receptor signaling and all. As you can see, a particular insulin receptor has been spotted on these muscle cells, and we are looking at the radiation scenario as well, a series of doses. So to summarize, metabolomic profiling of HSK-MCs showed a higher detection of lactate upon LDR exposure. LDR induces changes in histone modifications, and mass spectrometry profiling metabolomic profiling suggested lactate's involvement in histone acetylation. In vitro, in vivo differentiation showed a better treatment, or a better effect. In vivo regeneration was unaffected by LDR, but we see an enhanced myogenesis in that particular experimental setup, and fibers were bigger in size in the irradiated treatment group. And LDR exposure may delay the onset of muscle aging, probably through improved insulin signaling. Molecular mechanism, possible reinforcement of epigenetic memory by lactate. That's what we imply in here, okay? Then the next aspect what we looked at, briefly I can go through, we looked at genomic instability marker within the histone modifications. And we compared, along with muscle stem cells, we included several other cell types as well. So muscle cells was compared with human cardiomyocyte cells, HCMs, and human small airway epithelial cells. So we picked these cells based on sheer radiation sensitivity. As we know, human small airway epithelial cells are known to be extremely radiation sensitive. And these group of cells are actually isolated from the bronchial tracheal region, and it has got the limbal mucus producing cells and alveolar cells, okay? It's a mixed culture of primary cells. So what we did was we exposed these cells to radiation, ionizing radiation from a cobalt-60 source, one single acute exposure of 100 milligray. And as you can see, in muscle cells, exposed scenario, you see an enhancement of histone modifications, basically implicated in genomic instability. And these biomarkers, these histone biomarkers are actually required for the chromosome integrity, okay? Chromosome integrity. And these markers were upregulated in muscle stem cells exposed to 100 milligray. But at the same time, we don't see a hell lot of a difference in HCMs, that's human cardiomyocytes. but we see a greater difference in cells exposed to 100 milligray in the small airway epithelial cells. That kind of pretty curious, and we decided to follow up further. And we looked at, human small airway epithelials in particular, looked at the expression pattern of histone modifications 6-hour and 72-hour post-radiation scenarios, okay? And as you can see here, acetylation at large kind of disappeared or reduced when you increase the dose from 100 milligray to 2000 milligray. And histone H3 acetylation in particular was kind of again further downregulated when you increase the dose. And histone genomic instability biomarkers also showed a similar pattern, okay? Then at 72 hours post-radiation exposure, single acute, we see a consistent downregulation of most of the genomic instability marker in lung cells, okay? So this is something worth to proceed and investigate down the line to look at, is there any additional biomarkers of histone can be used for emergency response or medical countermeasure triage. That's something I'm exploring with the company Active Motive. It's a US-based company, an organization, and we put together a grant proposal for NIH, Small Business Investigation Grant, SBIR, and also a bunch of grant proposals were actually submitted, Canadian Safety and Security from Government of Canada, then Northern Atlantic Treaty Organization, NATO, Science and Technology Grant, and Canada Owners Group. So all these grant proposals are actually under consideration to investigate or find new histone biomarkers that can be used along with gamma H2AX for emergency triage. And we are actually, earned a lot of progress in the last several months, and the company Active Motive is very much interested in developing a lateral flow assay in order to, execute further diversification in this particular area of research. And that's it. And I would like to acknowledge my organization and the team at Canadian Nuclear Laboratories research team and the facility, and we have a consultant at Institute Pasteur Shargim Tajbaksh, my collaborator at University of Ottawa, Dr. Jeff Dilworth, Mary Ellen Harper. She specifically helped me with metabolomics, and Jeff Dilworth specifically contributed towards chip sequencing cut and tag analysis, and funding Atomic Energy of Canada Limited, Federal Science and Technology funding. So that's everything, and that's it, and I would like to have questions that you may have with regard to my presentation. That's everything. Thank you so much.
So this is a wonderful talk, and now the open for question.
Okay, yeah, go ahead. Good morning, Sebastian. Interesting lecture here. As a practicing radiation oncologist, we always protect the cardia and from radiation whenever we treat the thorax, and particularly the breast tissue whenever we treat. If a low dose radiation is giving the benefit of a regeneration of the muscle tissue, why are we doing that? Why are we really taking so much of a care and to protect the cardia from the radiation, even as low as only 5 gray or even lesser? Don't you think it should be beneficial, and even if the small amount of radiation gets into the cardia?
So my data, I speak on the basis of my qualitative data set, right? Qualitative data set. But we need to actually take a very conservative approach, and we need to talk on the basis of radiation protection, right? Okay, so biologically speaking, right, cardiomyocytes are moderately radioresistant. Moderately radioresistant. When you compared it with skeletal muscle has an amazing regenerative capacity that is lacking in cardiac. So whatever muscle you have, that's going to be there for your lifetime. And you need to take a conservative approach. And we need to continue protecting heart tissue from any radiation exposure. And its function is so crucial, as we know.
Yeah, no, that's right. The point is well taken. That's in fact that's the reason why and we do that. But when I see your work, radiation protection of the cardiac, and to that extent, is it really necessary? The cardiac tissues are moderately radio resistant. So more so, we need not have to do so carefully to avoid that. Don't you think so?
Oh, say that again?
No,
kind of
no.
missed
The
that
cardiac
final part.
muscle tissue are relatively radio resistant.
Yeah.
So going all the way to protect the kind of cardiac muscle, is it really necessary as low dose as a 5 gray?
It's still necessary. The reason is being, even a slightest production of ROS can have effects in our heart. And you don't want to have a compromised muscle function in your heart, because that's the one and only organ pumping blood to the various part of your body. And you don't want to have a future consequences, latent health effects in the form of a myocardial infarction. That's something you need to avoid. But in the case of skeletal muscle, that's not the question at all. And we are not expecting any detrimental effect. Because of its nature, the stem cells are highly wired. That's the only explanation I can give you. And we need to continue to be very conservative with regard to heart tissue. And we can definitely discuss offline as well, okay? Thank you so much for the question.
Very interesting talk, Sergey. Thank you.
Thank you.
The association, the strong association you find with the insulin receptor and the recovery of the muscle by radiation raises the question as to whether by manipulating the receptor itself, we can achieve the same result without the need of complex equipment and also the concerns that are associated with radiation. That's the first question. And the second, I will add it and then you can go ahead answering, is the decrease in the histone acetylation patterns that you see with aging.
Yeah.
Okay? Do you know what regulates them?
That's an excellent question. So I'll take the first question, which is, we see an effect upon exposure to radiation, and we see the same effect upon exercise as well, right? Exercise produces ROS, exercise produces lactate in your muscle, low dose ionizing radiation exposure also do the same thing in there. So basically LDR exposure, I would consider this as an exercise mimetic, right? So whatever information you derive from your low dose studies can be basically those benefits can be derived from muscle exercise as well. The key thing is identifying the molecule who can do the trick. I think that is pretty much possible from low dose studies. At the same time, I mean, with regard to radiation protection aspects, skeletal muscle would take a backstage or backseat when it comes to priorities. That's the inference what I derive from this. Is that answer all the questions? Okay, thank you.
So, it's a very nice talk. Very
Thank you,
interesting.
Neil.
Two quick questions. The one thing is you have separate data sets, metabolomics, epigenetics, RNA
Right.
sequencing. So have you done any approach to integrate and to merge all this in the multi-omics analysis?
Absolutely. So I do have a lot of expression profiling performed, close to 8 TB worth of data. I haven't processed yet, And all of them are actually from type 1, type 2 unhealthy individual biopsy-derived stem cell exposed to a couple of doses. That being said, I also generated a lot of epigenomic profiling data by cut and tag. So obviously, integration of the omics approach is required, and I would definitely seek some of your opinion and suggestions as I progress.
Thank you.
Yeah.
Second quick question. We know that all these histone biomarkers
Right.
are very nice, but in the end it comes to a functional clarification. So what kind of modification has what functional impact or what functional readout in the end? So do any plans to test that? Because I think that is missing in actually all of the studies.
Right.
I mean, worldwide in all epigenetic studies, that we really go forward and connect the histone modification to the function.
Very good question. So just to address this particular question, I did an RNAi in human cells, human muscle cells, and mouse cells. H3.3, the major variant histone. If your cells doesn't have enough of H3.3 variant histone, the effect what I have seen from low dose, it's not going to happen. So I tested both systems and I confirmed that. At the same time, if you actually use trichostatin to stabilize acetylation and expose these cells to radiation exposure, then we see an enhanced myogenesis. So acetylation is required. If your acetylation on those critical gene sets at the promoter and enhancer region, if you can stabilize them, basically that's going to capture low dose effect more effectively. And your question is very wise enough. So we need to come up with a method to knock out or CRISPR-Cas9 based just suppress some of these machineries who actually makes the acetylation or these modifications in vivo, then obviously, yeah, that's another area where we can diversify further. And George has that particular question, aging dependent loss of this acetylation or any of this modification, what does it tell you? Obviously, the pros and cons with my experimental method or methodology what I use, these are actually 2D cultures, right? 2D cultures. Things would be maybe different if I use organoids, right? And the aging paradigm over there would be very different. But I see upon aging, there is a drastic reduction in certain histone modifications. But what does it do? Yet to be revealed. Thank you. Thank you so much for that question.
Thank you, Dr. Soji. Very nice talk, and a lot of correlation with my work, which I presented yesterday. That for protection, we are trying to induce glycolysis as well as metabolic modification. So my question is, as you mentioned in your answers, that these muscle cells are relatively more resistant to radiation. So why you have chosen muscle cells as a model? That is one thing I want to understand. Then second thing is as you said, that probably would like to check some of these markers, acetylation as a marker along with gamma S2X foci. So as you yourself said in your answer that during the exercise also, and so many stress, they also induce this kind of acetylation, and then how specific this marker would be? Thank you.
That is an excellent question. I think I may have to answer in 3 phases. Why did I choose skeletal muscle cells, right? So when I started this particular project at Canadian Nuclear Laboratories, when I was a postdoc, I pick and choose 4 different cell types. It took me a long time to establish hematopoietic stem cells, HSCs, which is again patient-derived, small airway epithelial cells, human cardiomyocytes. It took us a long time. And among them, muscle was the easiest. Ever since I had prior experience with muscle system for 20 years. My PhD, undergrad PhD, rest of, postdoc and all were in muscle. Again, that's again just a basic explanation, but the real reason is being, your body has around, I would say 40% of your body mass made of muscle. So in a radiation protection point of view, for nuclear energy workers who works in, nuclear energy industry, And if you consider Canada as a country, is actually driven by the most of the energy needs, I think 60% of the energy need in the province of Ontario is actually provided by nuclear energy. So that actually bringing things in perspectives, right? And there's a need to address what are the risks involved or nuclear energy workers are exposed to. So, and also, you know, your body mass, considerable amount is made of muscle. So what are the risks involved? And also, there are chances that radiation exposure can lead to the development of soft tissue sarcomas, but it is very rare. Given the fact that skeletal muscle cells are highly epigenetically wired, that's why it is very radio resistant. I think I gave you sufficient explanations.
Yeah. So this is last question from audience.
Thank you, chairpersons. My compliments to Dr. Soji for very nice, excellent deliberation.
Thank you. Thank you.
So, as we know that in space, in human space program, we have a challenge of maintaining the health of cardiac muscles. So do you foresee the implication of your research in space program?
Absolutely. I would say yes, many times. I'm actually in a program which is actually funded by European Space Agency called Icrr, Investigation of Biological Effects of Radiation. So I had particular experiments conducted at GSI, again funded by European Space Agency, a couple of years ago. I did look at the effects of space ionizing radiation on cardiomyocytes, and I performed several gene expression analysis and also histone profiling. I did notice that several of histone biomarkers kind of disappear upon radiation exposure, ionizing particle ionizing radiation. Definitely yes, there are a lot of implications, and also one more thing, combination of microgravity and radiation exposure. It has got a very vivid effect, I would say vivid effect. And the main aim was to actually uncouple or decouple microgravity from space ionizing radiation. That was the main goal of my work, which was committed or funded by ESA a couple of years ago. Again, this much information I can give you, but you know, we can definitely discuss offline. Yeah.
Sir, may I ask a question?
I think first Dr. Mishra question.
It's a very wonderful talk, Dr. Soji.
Thank you so
I have
much.
two quick questions. First, it is regarding your histone biomarker, and
Sure.
I see the histone biomarkers are getting published, and this is a new area. But is there any data of specificity in your case? The sensitivity is very good, but is there any specificity data you have?
Specificity with regard to stem cell type, or?
Yes, it is to the particular kind of cross sensitivity. Sensitivity means it is simply to your cells of interest, but the same results comes to other kind of tissue, other type of cells as well. So that is your specificity. So is there any specificity data have you been seen? That is the first question. The second question is, we see that many epigenetic modifications are published in cancer, and as far as the muscle cells you are saying, the cells are like weird in terms of this epigenetic response. So have you done some sort of proteins, profiling apart from proteomic study, like some gold standard work like Western blotting or something like that, to some specific protein to validate your DNA level data?
So far, my analysis, endpoint analysis, kind of restricted up to histone modifications only, apart from some metabolomic profiling performed in muscle cells. And your question is it specific to stem cell types?
Yes.
I would definitely reckon that. So when I compared human cardiomyocyte, human lung cells with the human skeletal muscle exposed to 100 milligray, I see tremendous changes in genomic instability biomarkers. Okay, genomic instability histone biomarkers, a total disappearance in lung cells. The reason is being it is very radiosensitive. So that is a specific response.
And for the second question, have you done any proteomic?
Proteomic profiling, no, I haven't done that yet, but eventually I will.
Yes.
All depend on, I must tell you this, the research priorities at Canadian Nuclear Laboratories is being set by stakeholders, federal stakeholders. So every single year, what they want, basically you have to align your work towards that. So that's the main.
That is a good point.
So
I have
next question by Dr. Professor Misra, I think.
I'll discuss
You can discuss
offline.
during the tea time or lunch. Yeah?
Thank you so
Please.
much.
Yes, yes.
Is it okay? I withdraw
Yes,
my
yes,
question.
it is okay, okay.
Oh, fine. No. Okay. So, this question is regarding your results on the aged skeleton muscle.
Aging,
And-
right?
Yes.
Yeah.
Yes, I saw the.
Yeah,
picture.
that was an in vitro
and then
model.
5 milligray then you give is better, improved now the signal, even better than the normal. Are you saying that from the radiation protection point of view, there will be a threshold of the radiation dose which should be now considered? The concept that lowest dose is harmful has to be done away with in view of your result?
That's a very good question. It is pertinent to radiation protection for sure. I can speak only on the basis of my qualitative quantified data per se. 5 milligray compared to 10 milligray, will it be more beneficial? So I would basically stay away from dose response. I would blanket, my statement is going to be a blanket statement. Below 100 milligray, this is what I see in skeletal muscle cells. There could be an enhanced effect if you try 50 milligray as opposed to 5 milligray. But that experiment has to be done, right? So without looking at the data, I can't say. Even a 5 milligray single acute exposure, I need to do the experiment and see. Definitely there is a finite window in which you may see the stimulatory effect. That's what I can say.
Thank you very much.
Thank you for the question.
So, Dr. Soji, I have privilege to ask something, okay?
Sure.
So I congratulate you first, and a lot of discussion gone on the as this is tyrosine modification, acetylation of histones and everything, and you wonderfully showed, okay? Answer. You see, as you said, you using a model cells, skeletal muscles is best as compared to other one, and you said that lactate accumulation is occurs initially by. So I just curious to know whether you have checked the hypoxia-like conditions or oxygen concentrations, one thing, or and mitochondrial membrane potential. Because already question comes from ROS and others, so these two things I want to know.
Absolutely. I think I have received these very similar comments from my internal peer reviewers before I put my manuscript. Mitochondrial potential and bioenergetics, and the other aspects. I think, can you just mention that again? The first
Hypoxia.
Hypoxia, right, hypoxia. So I know that hypoxia, when you culture cells in hypoxic condition, low glucose condition, you seems to have lactate accumulation in certain fibroblast cell types. In my case, if you ask me, did I try hypoxia? I would say yes, because my experimental model is such a way that I take biopsy-derived cells, put them in methyl cellulose-based hydrogel, and my cells are actually in suspension with a low oxygen, right? And I expose these cultures to radiation and perform the downstream endpoint analysis. So cells are actually in hypoxia because they are in methylcellulose with the full complement of serum with no adhesion. So cells are actually in quiescence, G0 state of the cell cycle.
Can I give one short question?
Sure.
So do you think it's possible to treat sarcopenia of elderly people by your finding, or LDR in combination with insulin and lactate administered, or is it possible, do you think? Sarcopenia could be the very important problem in elderly people for loss of muscle, yeah? If you can regenerate elderly muscle stem cell by
Right.
this
Professor Tashiro, thank you so much for that question. In elderly people suffering from sarcopenia, which is the muscle aging, again, it's a multifaceted issue. Also, insulin signaling and insulin pathways have vivid effect in these particular situations. But if you think that there can be an improvement based on exercise, definitely ROS generated stress, right? And as a result, lactate functionality and rest of the other stuff possibly improve the situation. But I'm not sure about components of insulin pathway and how different they actually behave in vivo model in the context of this aging.
So thank you very much for very fantastic talk and very nice discussion. Thank you very much.
Thank you for the opportunity, and thank you for chairing the session.
So now for the next speaker, I can invite the Niels Cordes from the Germany, and his title, the pan growth receptor inhibitions induce the EMT based resistant and associated with the clinical outcome in head and neck cancer, sarcoma cancer cells. So
Thank
yeah,
you.
you can introduce yourselves because I don't have here anything. So I'm sorry for that. Thank you. Please start.
It's fine. Thank you very much for the introduction, and it's great to be here again. And I really like to thank the organizers for inviting me and giving me the opportunity to share some of our data, which we recently published in molecular cancer. But before I start, I think when we look at the field of cancer research, then there are so many questions that are being asked, and really great diversity, and also looking at metastasis and circulating cancer stem cells and all these different aspects. But from my point of view, also basically being a physician, I think we did not understand the primaries. So why in the first step should we go beyond? No question that all these things are of great importance. But if we do not understand the primary tumor, so how can we optimize the treatment? And from that point of view, we have different things to look at. And these are very different. There's sensitivity to therapy, there's resistance to therapy, and resistance by therapy. And we need to look at all these aspects in a very differential manner when we want to better understand the heterogeneity of the tumor microenvironment, all these aspects which are important for the primary, and also for optimization of therapy in the way of personalized medicine. So when we look at this, it is still a very big clinical challenge, because actually in none of the tumor types this has been solved. And there are different impacts on these endpoints, and not only the genetic endpoint which we followed, 3 decades or even 4 decades, but there's this new era of epigenetics. We know that there are metabolomic influences, proteomic landscape is important, but also the stiffness of the tumor plays a role, and of course, what are we doing with our therapy? How does this lead to adaptive responses? And is there, I think there is a great opportunity, great window of opportunity to optimize therapy during therapy. So when we look at the personalized medicine and we have 3 people or a bunch of people with same tumor type, then of course we know that when we look in more detail, these tumors are very different. So we have an intertumoral heterogeneity. And at the moment, we give 1 pill to everybody, including radiation, and we know that there are different outcomes. So this is obviously not satisfactory, but what should we do? And of course, there are many approaches and many efforts to solve this problem, but in the end, I think we are not doing so well at this moment. So on the one hand, we set all our efforts in the last decades on molecular profiles. And of course, that gave us some insights, but most of the very specific drugs based on the genetics failed in the big tumor types. So my approach is that we need to consider massively the response patterns. So taking out the biopsy and looking what is the patient really responding to, that we eventually can adapt the tumor, the treatment, so individual treating concepts to improve therapy outcome. So for me and my group in the last 2 decades, my group focused on integrins, cell adhesion molecules, and receptor tyrosine kinases. And in quite a number of studies we have published, that we follow the concept that we have a single or double targeting of integrins plus receptor tyrosine kinases. Why? Because they are very upstream in the signaling network, so we can, if we target them, we can deactivate a lot of the pro-survivor signaling. And we also know that not only at the cell membrane level, but also in the cytoplasm, there are mutual and cooperative interactions that are pro-survival and really firing the tumor progression and development. So when we look at the focal adhesions to set the scene, so we know where we are, then we have the cells, and you can see here in these different stainings, I hope you can see it, EGF receptor and beta-1 integrin, which are at specific transmembrane locations called focal adhesions. And importantly, as you can see here, they are coalescing at this focal adhesions in a very tightly. Yeah? So there are mutual interactions between these receptors, and they have strong impact and pro-survival downstream signaling. And we know from the work of my group and many other groups that if we target these focal adhesions, either integrins or also other associated proteins, that we can radio-chemosensitize different cancer cells from different tumor entities to radiation and chemotherapy. So in this particular work, we selected a very important integrin, which is beta-1 integrin. And why? Because out of the 24 known integrins, it is forming 12. So it means that if we target beta-1 integrin, we can deactivate a tremendous part of the downstream signaling in the cytoplasm. And the selected RTKs, we have 10 because we know that there are FDA-approved drugs for these RTKs. So eventually we wanted to see, is there radiosensitization or radiochemosensitization when we approach these receptors, overcoming therapy resistance. And another aspect I really like to point out, and I have done this in the last meeting, but I think it's of utmost importance that we look at in which conditions we grow the cells. So 3D is obviously important, and we always happy when we see the cells in 2D because we can nicely see everything stained for any kind of protein. See, all the cells are spread and they look healthy. But when we compare this, in this case, with head and neck cancer tissue, then you see that when we stain for actin, the cells look completely different. They're round in most cases, and this means there is a huge difference comparing the in vivo situation and the 2D situation. And easily, you can embed the cells in 3D matrix, different kinds. In this case, it is matrigel, laminin-rich extracellular matrix. And you can see when you stain the cells, they have the same morphology as the cells in the tissue. Very, very important, because the biochemical cues and the mechanotransductive cues, they control the whole scenario, the genotype, all the phenotypes, and so they eventually control the behavior of the cells. So if there is a difference in the repertoire of proteins and kinases and adaptive proteins, then of course the cells react in a different way than if the cells has another repertoire of proteins. And we know that the 3D growth really changes the possibilities of the cells on all these levels. So eventually, we know that if we take out cells and do not plate them in a 3D environment, and I do not mean suspension, I mean in matrix, then we have a loss of phenotype up to 90%. So setting the scene, I'm coming to the study we have done, and this is work of one of my most talented PhD students I ever had, Felix Burkhammer, and as the title says, it's about FGF receptor in HPV negative head and neck cancers. So in general, we know that HNSCC is among the top 25 tumors worldwide, and the importance here is that the efficacy of treatment has not really changed over the last 30 years. So that's still tumor type of unmet need, in particular also because of this therapy-limiting side effects, as you all know, working in the field of radiation oncology. So when we started the study, we looked at the beta-1 integrin and the outlined RTKs for mRNA profiling in the TCGA cohort. And as you can see, some of them, like beta-1 integrin and others, are significantly upregulated, others have no change. And so just looking at this and maybe coming back to this later and connecting the responsiveness, I show this data set. And when we look at the mutational landscape, you see among all that they are not very much mutated except FGF receptor 1. So the first step we have performed is making a knockdown screen in 3D grown HNSCC cells and looking at either cell viability or clonogenic survival. And what you can see here, this is the enhancement ratio. So every value above 1 is enhancing the radiochemotherapy we have applied here. And we did either a single knockdown or a double knockdown, always in the combination beta-1 integrin with 1 RTK. And as you can see here, there's a waterfall plot, and already when we have no radiation, we have a cytotoxic effect, for example, here in this case of FGF receptor 3. When we combine this now with X-ray radiation, because this was more or less a screen, then you can see also the different combinations, as expected, lead to different outcomes. And that we have done in the first part for 4 HNSCC cell lines. And what we saw is that FGF receptors were the main targets here, and when we inhibit them, they were able to enhance the cytotoxicity or radiosensitivity tremendously, also independent of beta-1 integrin. That was very interesting to us because all the other receptors we have investigated were very much independent of beta-1 integrin. So in the second step, to be more translational, we used a drug, edifitinib, plus cisplatin and radiation and an inhibitory antibody for beta-1 integrin to have a more, as I said, translational approach. And we used a set of 20 plus HNSCC models grown in 3D. And you can see here again the enhancement ratios. We could identify a responder group, an intermediate group, and a non-responding group, which is really covering the variety or a big spectrum, either with regard to intracellular or intertumoral heterogeneity, or with respect to inter-tumor heterogeneity. So using these numbers of models is of course very important to come to a strong conclusion. So from this we took 4 models, so 2 responding model and 2 non-responding model for our further research. And important here is that we now wanted to define it in more detail, the responsiveness to either beta-1 integrin inhibition or the edifitinib or other FGF receptor inhibitors. And interestingly, and this is really, I think, the most important finding of the whole set, is that when we look on cell viability, we have the responding cells which show a dose-dependent decrease in cytotoxicity and cell viability. But the non-responders were differently responding. So dose-dependently, we had a cytoprotection, which was unexpected because the cells were already very unresponsive. The same we could see when we add radiation. So the responding models were showing a decrease in cell viability, but the non-responding showed even a radioprotective effect upon the treatment with FGF receptor inhibitors. So of course we were interested in the mechanisms and did an RNA sequencing analysis in a responding model and a non-responding model after this treatment. And what you can see here from this heat map is that the responder and the non-responder models are very different. I only show here the top 5,000 genes differentially expressed, but we can say that basically and upon treatment, these cell types respond very, very differently. When we look at this dot plot here for enrichment analysis, then you can see that the responding cells have an enrichment in wound healing, tissue migration, cell adhesion molecules, something like this. And the non-responding cells have a pretty different pattern, like ion channels are induced and CMP signaling pathways. So that was basically the first analysis, and then we went and looked what happens after therapy. So we have FGF receptor inhibition alone, and then FGF receptor plus radiation. And we could see, and you can see this in this little graph here, when we look at the FGF receptor signaling alone, we have a reduction with the FGF receptor inhibitor in the responding cells, but an induction in the resistant cell model. So looking again at enrichment analysis, you can see that when we treat the cells with FGF receptor inhibitor, we have a decrease in many of these analyzed signaling pathways. So particularly when you look at the right side here, we have a suppression of many signaling pathways, which is PA3-AKT, which is focal adhesions, and so on. So it means there is a strong deactivation. But when we look now at the resistant models, we have a completely different pattern. So this is this model here. When we add the inhibitor, we have an induction. We have a really stimulation of all these pro-survivor signaling pathways, and on the right side you see that there's hardly anything suppressed. So what does that mean? When we look further, we saw with different bioinformatic analysis that particularly the epithelial to mesenchymal transition is induced associated with MEP kinase and EGF receptor signaling. So that prompted us then to look more into detail, and looking at the EMT process. As we know, this is a process present in many of the solid tumors. And it's not so much the polar opposites of this transition that are critical for therapy resistance and therapy outcome, but particularly the intermediate states, the partial EMT. And there are many groups who have worked on this, and this is very clear that this intermediate stage is inducing collective migration, is inducing plasticity. So all the things we like to avoid, but in this case, we have induced this state with our treatment. So that's why I want to emphasize that our investigations upon therapy, during therapy, is so important for the optimization of therapy. So the next step was that we wanted to do something with the data in a translational manner and see what are the real vulnerabilities. You know, we have the omics data, which is all nice. We did all this bioinformatic analysis, but can we do something with it? Can we translate this to really more effective treatments and to overcome the cytoprotection and radioprotection induced by FGF receptor inhibitors? And what you can see here is that we took the omics data, screened the first 500 or the top 500 induced genes for druggable targets. Of course, that is important, the druggable targets. And we found quite a number of them. And in this case, I show you the data of 35 inhibitors we have used and what the outcome is. So on this axis here, you see the single inhibitor efficacy. And in this part here, you see the combination of FGF receptor inhibitor plus the additional kinase inhibitor. And what we want to see is, we want to see if, when we add the second inhibitor, the cells are sensitized, have less viability. So what is important here is the lower left area here, what you want to look at. We come from here, this is the cytoprotection, and what is now moving the cell viability down here. So, and the same we did, of course, in combination with radiation, also the radioprotection, which inhibitor is able to reduce this radioprotective effect induced by FGF receptor inhibitor. And in the end, to summarize this, from the 34 inhibitors, many of them were effective, some only for the cytoprotection, others for the radioprotection. In the end, 13 kinases in the middle here shown can or led to a really significant decrease in the cyto and radioprotection we have seen in the resistant models. And then we translated this, checked also together with Hans Klevers in HNSCC organoids. We found the same response pattern in HNSCC organoids, which we have found for FGF receptor inhibitors in the more established cell models I have shown you. And also, we have tested the second inhibitors, the combination with the second inhibitors, and also saw that taken from fresh tissue, we can sensitize these cells and select patients that are better with 2 inhibitors than with 1. But the other thing we have done is, can we now take this EMT signature we have found and combine this with clinical data in the setup to eventually come up with a gene signature and a network analysis. So we took the TCGA data, and as you can see here for overall survival and progression-free survival, that when we apply the signature, we can clearly differentiate the patients which have a benefit and could be pre-selected for the signature, and others who are doing worse. And when we combine this with the cohort on then we find a trend which is not so significant when we look at the overall patient survival. But we will follow up on this, and of course it needs a validation study. Although we have taken data from Fred Hutchinson Cancer Center and MD Anderson Cancer Center and showed the same thing as from the TCGA data. So there's a strong strong signature that we can use in the future to differentiate and maybe to stratify patients. Another thing we have done, which is I think of utmost importance, is to see that the TCGA data is taken from biopsies, so we don't know how many tumor cells are in these biopsies, or what the pattern is we have found. Is this immune cells, activated fibroblasts, endothelial cells, or really tumor cells? And that's why we added a single cell RNA sequencing analysis on this, and we found, interestingly and nicely, we found the same signature in the single cell analysis as we have done for the bulk. This is very promising. And in the network analysis, we have done, using different databases, String and Gmania mainly, that the driver here is the EGF receptor pathway. EGF receptor, together with other downstream signaling components of this network, that is driving the EMT process in head and neck cancers when we apply FGF receptor inhibitors. And I think that's an important finding, that we see we cannot just take any RTK and inhibit it and then hope that it will work in the clinic. So what did I show you? That pan-FGF receptor inhibition plus minus beta-1 integrin inhibition radiocchemosensitizes HNSCC cells, but on the other hand, it leads in a subset of models and also organoids, as I said, to cyto and radio protection. And this is very alerting, I think, and we need to look in more detail how can we select the patients. And this has been then followed through by us identifying a partial EMT transcriptional profile. The good thing is we also provide data, for specific data for additional pharmacological inhibitors you can use to avoid or to overcome this cyto radio resistance induced by an inhibitor. And I think that can be also done similarly for other molecular targeted approaches. And what is nice here, that we found that this is associated with clinical outcome, and we can come up with a gene signature that now needs to be validated in prospective clinical trials. So I started with this idea here, and of course the main topic was resistance to therapy, but we can also see that not only to therapy but also resistance by therapy is induced, in this case FGF receptor inhibition. And we need, from my point of view, we need to tackle this problem and have more investigations of patients during therapy. So not only the molecular profiles are important, but to take biopsies before the patient gets the first treatment, make a responsive profile for promising small molecules, and then we can have an optimization upfront, and hopefully that leads to better outcome. So with this, I'd like to thank all the collaborators. As I mentioned, Hans Klevers and his group. There's also particular people who assisted us with the bioinformatics, people who gave us some more models to expand our panel, and there are a lot of current and former students that supported the studies in the last couple of years. Also a lot of funding agencies, and I thank you for your attention and happy to take questions.
Yeah, open for questions.
Excellent talk, Niels. Thank you. What I found interesting, and I was wondering what you think about it. Your inhibitor treatment profiles do not change whether you add radiation or not. You may get a little bit bigger numbers, but actually the treatment profiles more or less remain the same. Also, I think it's interesting that when you did your gene enrichment profiles, you find all sorts of genes, but you don't find DNA damage response genes. They didn't feature, at least in the graphs that you show prominently. I thought, I'm sure you have thought what this means for this kind of combinations. And the last issue that I was wondering is, you use everywhere 6 gray, which is, relatively speaking, in a radiation therapy setting, a high dose. Do you see similar responses also at low doses?
Thank you, Georg, for the questions. We have done, I only showed you the main data, and we used the 6 gray to have strong responses. We also did that 2, 4, 6 clonogenic assays and other response profiles. You can look that up in the paper. And we see the same thing. Yeah, so they're, of course, lower or smaller, but the same trend. Why, my interpretation is why we did not see a lot of DNA repair proteins is because this signature is so prominent. It's the only thing I can say. So it does not really, the radiation does not add a lot to they have gained some capabilities to deal with it even better. The first question was
So, Dr. Niels, this very enlightening and very advanced level of understanding that you deliberated here. I'm little curious to know that why you did not disclose the EMT-related kinase inhibitors. It potentiated, right, the FGFR therapy in combination. So would you like to specify some of those kinase inhibitors which enhanced the effect of FGFR therapy?
As you saw, yeah, thank you very much, Ahmed, for this question. As you saw in my one slide, there are all the inhibitors mentioned.
Okay.
Yeah, so they're also on the paper.
Okay.
Yeah, we had some EGF receptor inhibitors, also downstream of EGF receptors. So many targets which we have found in the omics analysis, we targeted because they're druggable targets. And also the data with the inhibitors is directly in line with the omics data. So the signature we find, the genes in the signature are the same or very much overlapping with inhibitors we have used and which are able to overcome this resistance.
Okay.
Yeah. I mean, there are also others, yeah, yes. Because also other inhibitors were effective. Yes. Some expected, some unexpected.
Yeah. So, can I continue just one more small query? So, as you know, the tumor is highly heterogeneous and it is a radiation acquired phenotype, that partial EMT. It means not all tumor cells will achieve this phenotype at the same time after the radiotherapy. So do you think more research is required to time the EMT based drug in combination with FGFR?
Thank you.
Yes.
As you know, this is a difficult question. I think the importance is that that we know already that there could be an induction of resistance. So whether it may be the whole tumor or the majority of the tumor which is resistant already, or there are subpopulations, doesn't matter, or whether we induce it. So I think the status of the tumor before therapy can give us this large window of opportunity to optimize already there, because we know, and what we have done here is that we treated the cells with inhibitor plus minus radiation, waited like 3 to 4 days. We have also done an experiment to look at 24 hours, and we already see the beginning of this EMT phenotype. So it means it would be a quick predictive assay. And of course, in the best of the worlds, it would be great to, if you have the 6-week time course, for example, for head and neck cancer, radio chemotherapy, that we look at 2 weeks or 3 weeks and see how did the tumor adapt, and adapt or adjust our treatment accordingly.
Oh, yes.
So, yeah.
Yeah, nice. Oh, yes.
Dr. Soji, I think first.
It's a great talk. In the context of precision or personalized medicine, which you nicely described, your talk also kind of begs a question, important question, that you talked about biopsied samples from tumors. You also talked about cells circulating, which we call as CTCs, circulating tumor cells. So CTCs are a dynamic model, whereas tumors from a particular tissue, you might call them as a static model. Okay. Now, the CTCs are susceptible for lots of changes. They have to sustain continuous re-education by macrophages. Even the T cells get re-educated to become, for tumor and all that. That's all part of the CTCs. Whereas the tumor tissue, which is a static tumor, it may not go through that kind of a harsh reality. So, keeping these two scenarios, how would you envision what you showed, the precision medicine? You talked about heterogeneity and all that, but heterogeneity is not discussed in terms of these two scenarios. It's mostly discussed keeping the static, the tumor tissue in mind.
Thank you for the question. I believe that the same approach is targeting the circulating cancer stem cells, or whatever the subpopulation is, because these are part of the partial EMT, or this intermediate stage where the tumor spreads massively and is resistant or becomes resistant. So, so I think when we know about the primary, and the next step, of course, as you say, would be interesting to apply this also to CTCs, whether it's similar effective, but I believe it will be. And I think that we, as I said, we did not understand neither the heterogeneity nor the dynamic after start of treatment. Yeah, I think that is that's a huge gap. That there's very, very few groups who have looked into this, but I think we need to overcome. And in most cases, I think, at least for Germany, there's always this ethic argument. But I think this does not help us. So we need more studies looking at all these aspects during therapy, including liquid biopsies, including circulating tumor stem cells.
Thank you, Professor Neil Cordes. Excellent talk. I'm trying to lay out a couple of contentious issues, which is, I feel it's okay to discuss, which is
Please.
Tumor, some of the contentious issues in cancer radiation therapy, right? Tumor radioresistance, driven by EGFR, FGFR receptor cooperations. The question is, how often are we sure the substrate kinase interaction in this particular setup? And can we use AI-driven CRISPR-Cas9 targeting of these kinases to actually bring down, or by a moonshot, you can actually suppress the kinase activity just to alleviate radiation resistance? And is it possible to actually find a common kinase inhibitor as a comprehensive inhibitor for this treatment?
Thank you for the question. I understand that you talk in the context of the clinic, yeah? That's right. Yeah. I think that the biggest challenge is bringing, if you have a CRISPR or a knockout approach, to deliver it to the tumor cells. You know, how specific can that be? And I think there are not very promising studies out there. At least at the moment, I don't know any. And I think if we talk about vaccines or this, I'm not so positive about this, I must say, because we still have the side effects. We still are not so specific with delivering neither mRNAs or whatever to the tumor cells, or to this conglomerate of what we call a tumor. And I think, yeah, so that is my impression about this. And to have the ideal RTK inhibitor, it needs that we characterize more before therapy. I stay with this concept because we know the intra and intertumoral heterogeneity. Whatever that comes from, yeah, we don't know. And if we can also detect this in liquid biopsies, and I think this would be the most easiest and elegant approach because we don't need to put a needle in the tumor, I'm not sure. And from my point of view, there's a big lack of integrating all this data we have. So many I don't know how many million cancer researchers we are, yeah, but to integrate the knowledge we have with AI or machine learning stuff would be a great benefit, and then I think we would see that we have much more than we think at the moment. But as these are single data sets, we cannot really combine. I think that would be the way to go for your last part, whether there is like a common RTK approach, anti-RTK approach or something.
In the current clinical setting, we do use EGFR assay, that we do it, and we do anti-EGFR treatment along with radiation. That's common. If there's a synergy in between EGFR and FGFR, is it necessary, that to still go ahead and with the, pan FGFR assays, or just what we are doing is quite good enough? That's point number one question. Number two, the circulating...
I did not get the question.
Yeah, no. The question is, in the clinical scenario that we do today, anti-EGFR treatment along with radiation, we practice.
Yes.
So when we are doing it, still is there a need to go ahead with assaying, FGFR, and because I see the synergy in between the two. That's my, got it? Okay, that's one. The second one is regarding the circulating tumor cells. Today we use it as a more as a surveillance issue rather than to treat the cancer patients. More on a surveillance, how the disease are really progressing and how do we really use it of surveillance, not today. So therefore, is it still necessary to again use the circulating tumor cells also in these in these scenarios? This.
Yeah, I start with the second one. I think it. I think it's important to address this question, how we can target the CTCs, because we think that they lead to metastasis, and of course the only reduction of metastasis or the intravessel agent we have is the chemo. Of course, radiation also can reduce metastasis, but I think to really have a better targeted agents for the CTCs to follow up this concept and treat HNSCC CTCs, for example, in a quick experiment, and to see if this is effective would be very interesting, because then we see that the combination also is important to not give locally, for example, which we also could think of, but this is a systemic treatment. And the first part you had is, well, again, it is complex and not so easy, I think, because when we studied the interaction of, for example, beta-1 integrin and EGF receptor, we saw that is a common pattern. So when we have a resistance to the one, we have a resistance to the other. Meaning that whatever we combine, we need to test beforehand. So if it would be beneficial to combine cetuximab radiation, for example, with an FGF receptor inhibitor, I think we need to test that for it, if that was your question in that sense. Yeah, I think we need to test it, and we can test it. It is doable. We and others have shown that, but it needs to be done. And I think that is the part of the clinic where we can say it is not always like just a drop of blood where we can see everything. Maybe we can at some day in the future, but at the moment we need to take a little bit of biopsy, test that, and the data, at least our data, shows that it is not so dependent on the localization where we take the biopsy, but is that we take the biopsy. So we have taken different biopsies from one tumor, and they respond very similar. Actually, from my point of view, very unexpected, because I thought there's more heterogeneity, but that we can do, and we need to test it. We need to do it.
Yeah. Dr. Pandey.
I think we are getting late but
question.
still I cannot resist to compliment you for your talk. And also I got one quick query, like you are studying this integrin, and also we are working in 3D models where maybe the cancer stem cells may be also playing role. So how these cancer stem cells in overall your all processes of your story, they are going to contribute in this scenario, especially in spheroid model or 3D models.
You're asking about 3D models?
Yeah, 3D. So where basically cancer stem cells may be more contributing in the terms of radio resistance or your radio protection.
I did not get the question.
Yeah, my question is that like in the 3D model, the cancer stem cells also may be there in your spheroid models. Some percentage may be about these cancer stem cells. So how they are going to play a role in the radio resistance feature of your story, integrin story? Yeah, Whether I could.
I still did not get the question.
Question is that in how this whole story, cancer stem cells are going to play a role. Since this is a 3D model, cancer stem cells, which are basically
cancer stem cells.
Yeah.
Ah. Oh, cancer stem cells. Yes, thank you. The cancer stem cells. I'd like to call them cancer initiating cells, but that's semantics, I guess, in the end. The cancer stem cells, of course, or this subpopulation is of importance. We see that in the clonogenic assays, for example. And we see that there is it is important also here when we investigate these cells that we keep them in 3D. Because, I mean, in the tissue, we think they're growing in a particular niche, and we can really modulate, or we can mimic this niche in the 3D context, but only if matrix is there. So the matrix is important because when we, for example, put these cells into 2D or into suspension, and we compare either transcriptome or kinome, we see that the response is 2D. So cells which grow in suspension, as spheroids, for example, they behave more like 2D grown cells than like 3D cells in matrix. And that's also true for the cancer stem cells. Yes. And I'm happy to discuss this further with you.
I think no more questions from the audience side. So Dr. Neil, you give wonderful talk and you cover the so many things using the genomics and pick up so many genes from the integrins and receptors, integrins, as well as the adjacent molecules, and then proof with the transchromatic data, okay? And you showed the co-cultured cell lines, and then go to the protein-protein interactions, okay? So I am just interesting to know the interaction, specific some proteins that involved in the adhesion. For example, that you can prove by using other technique, because string database that we using, that is hypothetical, okay, interaction given layers. And you can see so many protein interact with if you see the interaction, okay? So did you check to other technology or methods that can prove, okay, these EGFR or other specific integrin, beta integrin can interact to these molecules? So this type of things, did you check?
Yes, thank you. Yes, we have done this in previous studies, and we found that it is when we do an IP of an integrin or EGF receptor or FGF receptor, we see that in the complex. But we see also it is not a direct physical interaction, but there are several interacting proteins between the integrin, in this case beta 1, and the receptors. And that not only in the cell membrane, but also, for example, in vesicles. Because when you look at caveolae, for example, the integration, the invagination, yeah, then we find this interaction also constant constant in these vesicles. So also the signal transduction is going from the cell membrane down to the nucleus.
So the fold changes also occur as we can see in the transcriptome data. Did you check the fold changes?
The fold changes?
Upregulations, as we see in the transchromatic data.
Oh yes, yeah.
Okay, thank you.
Yeah. Can I have one question? So it's very short. So you show a very nice data concerning the relationship between EMT and FGFR pathway. So do you think it's this EGFR receptor signaling is downstream from EMT or the upstream or from your data? What could be the suggestion for this? EMT is regulated by FGFR system or EMT regulates?
Well, in our case, we saw that the inhibition of FGFRs is propelling, is further inducing this phenotype. It was already there in the basally, but it was then further induced. And interestingly, and I think it's just a switch to a different receptor, the cells gets the benefit for EGF receptor signaling instead of FGF receptor signaling. Of course, a lot of work needs to be done to really untangle what is hen and what is egg, but and I think it seems, and we found that there is a difference between the different FGF receptors. For example, 1 and 4 are much more important than 2 and 3. Yeah, so we did that with knockdown studies and knockout studies to see which FGF receptor is really the driver of this process.
Thank you very much.
Yeah.
Okay, thank you very much. Niels, very fantastic talk and very stimulating. Also, it stimulated so active discussion between audience. Thank you very much
Thank you
for
very
the
much.
very nice discussion. Thank you very much.
Thank you, Chairperson, and thank you, speakers, for valuable insights. To move on to our next session, tumor radiosensitization by targeted agents, and to chair the session, I would like to call upon stage Professor Avinash C. Pandey, Director, Inter-University Accelerator Center, Delhi, and Dr. Pritanjali Singh, Head of Radiation Oncology Department, Aiims, Patna. Please join us in welcoming them. And I request the chairperson to take over the session and invite the speakers. Also, I request that due to as we are running short of time, we request the chairperson to take control of the discussion timings after the lecture.
A very good morning. To start this session, first I would like to call upon our speaker, Dr. Ashwini Sood. He is well renowned and he is going to talk on the rational decooperation approaches for radioactive actinidines from mechanism to mitigation. And he has a research interest in radionuclide therapy, nuclear cardiology, general nuclear medicine, and he has a lot of papers in many distinguished journals. So over to Dr. Ashwini Sood for his very interesting talk. Thank you.
Thank you very much. Good morning, everybody. Probably there is a confusion in the title of the talk. My talk is Targeted Therapy, the New Era of Personalized Medicine. So I'm basically a clinical doctor, and I'll be talking about the
Please
therapy.
go on, yes.
Thank you. So without wasting time, we know that there is a tremendous development in the nuclear medicine for the diagnostic imaging followed with the radionuclide therapy. And we all know that the molecular abnormalities often precede anatomical changes with the various malignancies, and there is a lot of development that has taken place for the functional as well as the anatomical imaging in the form of a SPECT-CT, PET-CT, and PET-MRI, which can do a whole body imaging. And newer radionuclides and radiopharmaceuticals are also on the horizon. So theranostic is the future of a nuclear medicine, where same or similar radiopharmaceuticals that share a specific target in the disease cells or tissue, and they give us a diagnostic as well as a therapeutic purposes. So for imaging, we use gamma emission in the form of a technetium-99, iodine-123, gallium-68, F18, which gives us a diagnosis. And for therapy, we use beta emitter or alpha emitter, like iodine 131, lutetium 177, samarium, actinium 225, radium. So basically, they are bound with the specific molecule which targets the symporters or receptors at the cellular or subcellular level. So this is a list where we see the theranostic pairs like iodine 123, which is used for imaging, while iodine 131 is used for a thyroid cancer patients, like gallium 68 PSMA or F18 PSMA is used for imaging, while when it is labeled with the lutetium, which is a beta emitter, then it is used for therapeutic purposes. So starting with the birth of an idea, this news was published in 1940s when local researchers discovered that the cure of a goiter can be resulted with the atomic bomb byproducts. And this was the first person who was treated with, who was having a CA thyroid, and he was having a widespread skeletal metastasis, and he was treated with 4 cycles of radioactive iodine. And you can see the difference in the picture. This was the pre-treatment picture, and this was the after-treatment picture. So this patient was treated with the radioactive iodine. So the journey so far, in 1951, FDA first approved iodine 131 for the thyroid disease, and now up to 2018, NETTER-1 trial, which was published in NEJM, and on the basis of that, FDA approved the lutetium dotatate therapy in a neuroendocrine tumor, which is inoperable or metastatic disease. So this was the first trial that was known as a NETTER-1 trial, which was published in NEJM, and here more than 200 patients were taken, and it was done as a second-line treatment where lutetium dotatate therapy was given versus octreotide, which is still considered as a first-line treatment, but it showed that at 20 months cutoff, the PFS was 65% in a lutetium therapy versus 10.8% in a standard care of treatment. Response rate was also very high. Overall survival was also 48 versus 36 months. So in this case, lutetium therapy was given as a second line of a treatment. We published our data. We published our data in 2020, where we showed that lutetium therapy can be given as a first-line treatment instead of a second or third-line treatment. And we evaluated 45 patients. Out of that, 40 patients were analyzed, and we showed that almost 85% of the patient has disease control rate, and the median progression-free survival was 48 months. And we can see that also there was a significant difference response in the tumor. We can see over here, this was the pre-treatment and this is the post-treatment images. So we also publish our data where lutetium dotatate as a first-line treatment was compared with the octreotide, which is still taken as a first-line treatment, and we showed that the overall objective response rate was in a PRRT arm was 38% compared to just 15%. The disease control rate was also 88% versus 67. PFS was 54 months compared to 16 months. And we can see the difference in the survival in the lutetium dotatate therapy versus octreotide image. And this is NETTER-2 trial, which is recently published in Lancet, and here they have started using the octreotide lutetium therapy as a first line, and they have also showed that objective response rate was 65% versus 7%. This is the arm where the octreotide therapy was given. This is the arm where the octreotide plus lutetium dotatate therapy was given. Even the median PFS survival was 22.8 months versus 8.5 months. We also treated our patient of a medullary thyroid carcinoma where nothing was available, and these patients were progressing on this thing, have shown the progression on the other type of a treatment. We treated those patients with the octreotide plus low dose capecitabine, and out of 8 patients, 1 patient was lost to follow-up, 7 patients were evaluated, and 6 patients showed a stable disease, and 1 patient showed the progression of that disease. And we can see this patient has a mediastinal disease which is inoperable, and patient was not showing any response. The patient was treated with the 4 cycles of lutetium dotatate therapy, and we can see the disease has gone down, and patient was radiologically progression free up to 4 years. So coming to the prostate cancer, we all know that it has a natural history. Initially, whenever it is a localized, normally it is done with the prostate surgery or radiation, or patients are put on active surveillance or watchful waiting. But some of the patients, they'll still progress with this type of a treatment, and they may have a metastasis. Metastasis means the disease can go to the lymph node, skeletal, or visceral metastasis. So these patients are normally treated with the androgen deprivation therapy along with abiraterone. These are the basically abiraterone and enzalutamide are androgen receptor pathway inhibitor, or they are treated with the docetaxel. But in spite of that, it has been shown that they will progress to the metastatic CRPC, that is a castrate-resistant prostate cancer. And these are the patients, again, where these type of a treatment would be given to the patient, but they'll ultimately progress to the, they will fail to show the stabilization of the disease, and they'll progress. Now, PSMA-based therapy has come, and there are two trials which have given way to the PSMA therapy. That was a therapy trial and vision trial. So first one was the therapy trial, where roughly 200 patients were randomized into 1:1 ratio. One arm was given lutetium PSMA therapy, another arm was given a cabazitaxel, which is a taxane-based therapy, and it was shown that the primary outcome was the PSA response rate, and we can see in the cabazitaxel arm, the PSA reduction equal to more than 50% was just seen in 37% of a patient, while in lutetium arm it was 66% of a patient. However, when the overall survival was taken into account, it was almost similar, 19.1 month versus 19.6 months. So this was the landmark trial, which is known as a vision trial, and here almost more than 800 patients were taken into 2:1 ratio, and in the treatment arm, patients were given lutetium PSMA therapy plus standard of care treatment, while in the other arm, patients were given standard of care treatment. And we can see the progression-free survival was almost 8.7 months versus 3.4, while the overall survival was also more, it was 15.3 months versus 11.3. And this trial was done in the patients who were not showing any response to the existing treatment and all these things. Adverse events were slightly higher in the treatment arm, but they were manageable and they were not of much of a concern. So the conclusion of the vision trial was that the lutetium PSMA 671, when it is combined with the standard of care in the metastatic CRPC post ARP, means these patients have been treated with the androgen receptor pathway inhibitor as well as on the chemotherapy, and they are still showing the progression. They can extend the overall survival, it can delay the radiological progression-free survival, and lutetium PSMA 617 radioligand therapy is well tolerated, and it is a new treatment option in patients with metastatic CRPC. So on the basis of a vision trial, FDA approved this Pluvicto, which is a lutetium PSMA 617 approval for the metastatic castrate-resistant prostate cancer. So coming to that, what is next now? Because it has been shown that in spite of patient being treated with a lutetium PSMA therapy, ultimately they'll show a progression of the disease also. So one of my colleagues did a meta-analysis of 13 patients where it was shown that when lutetium PSMA therapy is given in the taxane-naive CRPC patients compared to taxane-treated patients, it has been shown the lutetium PSMA therapy will show a better response in form of a PSA response by 1.8 times better. Progression-free survival was also better in 40% of the patient. Better overall survival was also seen in the patients who were taxane-naive CRPC patients. So we also did one study at our center, which was a randomized control phase 2 non-inferiority trial, and it was published in EJNMI. And here in this case, we took two arms. One patient, they were treatment-naive patients, and in one arm, we gave them lutetium PSMA, and in another arm, docetaxel was given, and it was shown that the 50% PSA response rate was higher in PSMA arm compared to docetaxels. PFS was also higher in a PSA arm compared to the docetaxels, and the treatment-related adverse events were lower in PSA arm compared to the docetaxel. And we can see with the waterfall plot also, there was PSA response rate more than or equal to 50% was in 60% population of the PSMA compared to the 40% in the docetaxel arm. And we continued that study with the final survival, and it was published in JNM recently, and we found that the overall survival was almost same in both the arms. So this is, now we wanted to see what to do next, because most of the patients were progressing on the disease. So we published this case report in European Journal, where it was taken as a cover page also. And here we gave the lutetium PSMA short course therapy in the metastatic hormone sensitive prostate cancer. Means we prepone the sequence of the treatment, and this patient who had a high volume metastatic hormone sensitive prostate cancer with a PSA of 800, and we can see the patient had a skeletal mets and lot of high volume disease. And patient was given 2 cycles of PSMA, and patient showed a very good response. PSA came down to 0.25%, and patient was kept on follow-up. Ultimately, PSA came down to 0.12, and patient was followed up to 18 months. After 18 months, patient developed a skeletal mets in the left ischium, which was treated with the external beam radiotherapy, and patient is still surviving. So what to do now? Till now, lutetium therapy is recommended in the last stage, means third stage, as a second line or third line treatment. So now people are trying to shift or prepone the treatment of lutetium therapy as a first line treatment, and there are a lot of trials which are going on, and we have to see the results, but I will be discussing one of the trial which has been recently published. Now even the people are trying to use the lutetium therapy in a metastatic hormone sensitive prostate cancer also. And I showed you one case where it showed an excellent result. So this is the PSMA-4 phase 3 randomized control trial, which is recently published, and it showed that the patient who were treated with the lutetium-67 arm, and they showed a 51% response rate in a PSA, while other arm where abiraterone versus enzalutamide was changed, here the response rate was only 17%. Even the radiological progression-free survival was almost double. It was 11.6 months versus 5.5 months. So apart from that, it has been shown that whenever the patients are treated with the enzalutamide, it can increase the PSMA expression, and probably that can be used for betterment of the PSMA therapy also. And in this case also, we treated this patient, and this patient has a skeletal mets in the rib, in the pelvis, and in the bone. And these are the post-therapy images. This is the anterior image, and this is the posterior image. And we can see when the patients were treated, there is hardly any tracer uptake in the lesion. These are the bony lesions which are showing uptake. We gave him a short course of enzalutamide, and we can see there is an increased tracer uptake in the lesions. And we calculated the tumor parotate ratio also, and it increased 2 times to 3 times in a post-therapy image also. So enzalutamide can result into increased PSMA uptake also. And recently a trial has come, which is recently published in 2024 Lancet Oncology. Again, in this phase 2 trial, they took 162 metastatic CRP patients, where half of the patients were given enzalutamide plus lutetium, and another arm where the enzalutamide alone was given, and it showed that the patients who were treated with both lutetium as well as enzalutamide, they have a PSA progression-free survival of 13 months versus 7.8 months. The adverse events were almost similar, and the conclusion of the study was that a lutetium PSMA, when added to enzalutamide, can improve the PSA progression-free survival metastatic CRPC patients. So coming to till now I have spoken about the lutetium therapy. So we have a targeted alpha therapy also, and we know that alpha therapy in the form of actinium labeled PSMA or DOTA have shown a promising response rate in patient even with the disease progression after lutetium PSMA. Means they can still show efficacy to the patients when these patients have already failed with the lutetium PSMA therapy. It is because of alpha therapy has a short range and has a higher energy, so it improves the treatment efficacy, and it decreases the hematotoxicity in patient with the diffuse skeletal mets. We also published our initial data where we try to assess the quality of life in 11 patients. It was published way back And we showed these patients were already treated with the all type of a treatment available, including PSMA therapy. In spite of that, out of 11 patients, 5 patients showed a PSA response more than 50%, 3 patients showed a stable disease, while 3 patients showed a progression of disease. So these patients didn't have any option, but when we treated with the actinium PSMA therapy, they showed a better results. And we assessed the functional PSA symptom index also, and we can see the pre-therapy score was 29.8, which increased to 41.3. So we could say that their quality of life was also improved with this type of a treatment. So we also did a systemic review and meta-analysis from our department, where we try to see what actinium PSMA radioligand therapy can be useful. We took 10 studies, more than 250 patients were taken. 63% patients showed a PSA response more than 50%, 74% patients showed molecular response. Median progression-free survival and overall survival was 9.1 months and 12.8 months. So these were the patients who were not able to afford anything, but when they were treated with the actinium PSMA therapy, we could increase the overall survival as well as the progression-free survival. The most common adverse event was xerostomia. Grade 3 toxicity was just 1 to 20% in these patients. And this is a paper which is known as a warmth act also, which was also published in 2024. And we were also part of this study, where they did the multicentric retrospective study, where the patients were treated with the actinium PSMA therapy. 488 patients were taken. In this group, median progression-free survival was 7.9 months, median overall survival was 15.5 months, and the waterfall plot shows that 57% patient had a fall more than 50% in this group patients. So this is one case which I would like to show to you. This is the patient who came to us in a wheelchair, and patient is a known case of a neuroendocrine tumor rectum with a widespread skeletal metastasis. Patient was not able to walk and all this thing. So instead of giving a lutetium dotatate therapy, we thought of giving a actinium dotatate therapy because of the diffuse skeletal mets. After 4 cycles of a treatment, patient show a significant improvement. We further gave him 2 cycles of actinium-225, and patient showed a complete metabolic response, and patient is on follow-up after 2021, and he's basically veterinary surgeon, and he's doing actively his job and all these things. So till now, what we know that the actinium-225 PSMA therapy is quite safe, but it can result into nephrotoxicity, and we lost this patient, though this patient presented with the metastatic CRPC with the PSA of 47.1. This is the disease we can see over here. The patient was given 2 cycles of actinium PSMA therapy. With the excellent response, we can see there is hardly any activity or disease in this patient, but the patient developed renal toxicity after 3 months of the last cycle. The histopathology showed interstitial edema, tubular injury in the background, tubular atrophy, and we lost this patient because of the sepsis. So apart from that, I'll be just talking briefly about the new targets which we have been trying in our department, and I'll show you few of the cases because these studies are still going on. This is one case report which was published in EJNM as an image of the month. And we can see this is a 54-year-old female CA thyroid patient who was treated with the thyroidectomy post-reductive iodine and sorafenib. Patient showed a progressive disease. We can see there is a recurrence in the thyroid bed. There is a skeletal mets. There are bilateral lung mets and all this thing. We can see patient was treated with the RGD, lutetium dotatate RGD, which is a tripeptide, and it has a high affinity for the integrin, which is overexpressed in the tumor, and we can see there is a tracer uptake in that. And after treatment, patient, we can see that there is a remarkable improvement in the size and the avidity also, and patient got relief from the unbearable pain, and patient was alive for 4 years. So there is another agent which is CXCR4, that is a chemokine receptor, and this patient was referred to us because of the refractory atypical mediastinal carcinoid. Patient was inoperable, and we can see there is a disease in the mediastinum. We thought of treating him with the lutetium dotatate therapy, but as we see, there is hardly any uptake, or that is a less intense tracer uptake compared to liver. So this patient was not fit for dotatate therapy. So patient had a FDG PET also where it was showing intense tracer uptake. So patient was subjected to chemotherapy, but in spite of chemotherapy, patient showed a progression of the progression of the disease. Patient was again sent back to us for another type of treatment, and we did with the CXCR4, and we could show that there is an intense tracer uptake in the mediastinal disease, and the patient was potential target for the lutetium PSMA, lutetium CXCR4 therapy also. So this is another agent which is fibroblast activating protein, and this is a case report. This patient had a adrenal cortical carcinoma which was operated 4 years back, but patient had a recurrence at the surgical site as well as in the liver also. So, and patient was having a unbearable pain. So we did a pre-therapy gallium-68 FAPI-486 just to see whether it shows any avidity or not. It showed the avidity. So we went ahead with a low-dose lutetium therapy just to see whether that tracer is retained or not. And we did the imaging up to 10 days, and we showed that the tracer is being retained in the lesions. And we treated with this patient with a 100 millicurie of a lutetium FAPI-2286, and patient became stabilized, and patient pain also got relieved. So our goal should be that we need to add years to the life and life to the year, but we need to be clinical flexible. I would like to draw your attention to one case, which is a known case of a metastatic CRPC patient which received external beam radiotherapy, docetaxel, enzalutamide in 2016, but patient showed a progression of disease, and patient was referred to us in December 2017 with a widespread skeletal and lymph nodal metastasis. We gave him 4 cycles of lutetium PSMA therapy. Patient became stable, but we lost the patient due to COVID for 1.5 years. Again, patient came to us with a widespread skeletal metastasis. We treated him with a re-challenge therapy, means we gave 4 more cycles of lutetium therapy, and patient tolerated well. Patient showed response. For 1 year, patient showed a stable disease. Again, patient showed a progression of disease. We again treated him with 2 more cycles of a lutetium therapy. Patient got stable disease, but last month only patient developed cerebral metastasis, and patient had an intracerebral bleed, and we lost him last month only. So patient was alive for 7 months, 7 years. Normally these type of patients may live for 2 years at the most. So with that, I would like to conclude my talk. So targeted radionuclide therapy for malignancies have shown a high efficacy with limited toxicity, and it has a huge scope in earlier treatment lines. Combination with the conventional agent has to be assessed for benefit versus risk ratio, but there are certain challenges like supply issues and the cost that we have to address. With that, I would like to thank everybody. Thank you.
Thank you, Dr. Sood, for concluding in time. So we can take one question.
I am from Sriji Radionuclide Production Cyclotron, commercial one. IMT Manisar, how you got, actinium-225 from PSMA, from where?
We got it from Germany, ITG.
Okay. There is a, we purchase a solid state target, and we are thinking, how actinium-225 is produced. It is radium-226. Right. It is to be bombarded in cyclotron. It is possible in cyclotron only, not in reactor. So the problem is the radium-226. We gave radium-226 to various hospitals long ago, if you remember, because of the radon problem, leakage of the radomes, So the IAEA is trying to hold meeting, it will be held, next month, regarding how to get radium-226 tubes and needles from the hospital back to cyclotron, and dissolve, and how to dissolve, because some radon escape will be there. It requires very minute 2 milligram. In fact, I wrote a project when I was a student, regarding the leakage of radium tubes and needles. The radium sulfate, if it is radium sulfate inside, it is completely dehydrated. It is possible to dehydrate. But if it is radium chloride or radium bromide, some H2O will be there. And because of the radiation inside, because it is a sealed source hermetically, hydrogen and oxygen will be produced because of the dissociate of, H2O, because some water molecule is inside. So radium sulfate, there is no problem of radon leakage as such. But nobody understood what I wrote, So radium sulfate one should have not given back to BRC.
Thank you for that.
Thank you, sir, for your comments. So I guess there are no other questions from the audience. One, yeah, please.
May I ask? Okay. Thank you, Dr. Sood, for this excellent presentation. The question what I have is very specific. My question is, do you see tremendous impact of actinium-225 PSMA on bone marrow residing hematopoietic stem cells? Do you see some off-target effects in your patients?
Can you repeat your question, please?
Do you have effects on bone marrow residing hematopoietic stem cells from PSMA actinium-225?
You have a very good question. Basically, we have treated patients who have a widespread skeletal mets all along with the bone marrow, because the actinium as a alpha emitter has a very short range. So we expect the hematological toxicity would be very less. But one or two patients of our ultimately died because of the platelet thrombocytopenia, means they had a very low platelet counts, and ultimately these things come to the disease also. So probably that would be there, but we have not studied about that thing.
The reason is being, even though HSCs, which is hematopoietic stem cells, reside in the bone marrow in a hypoxic environment, and there is no oxygen effect there, right? Still, actinium and its energy deposition is possible there, right? Off-target effects. And it can have deleterious effect on HSCs and its progenies. And one more thing I just wanted to ask you, why did you decide to use DOTA rather than MACROPA for your radio RIC conjugate?
Basically, your question might be, basically I'm a clinician. Right. So whatever has been approved, we are using those things only. So DOTA has been approved for as a ligand, dotatate therapy or for the imaging also. So we have been using, but I'll not be the right person to answer your question. So basically I'm a clinician, so I don't know much about those chemistry, radiochemistry.
That's right, okay. Thank you for your response.
Thank you.
Next question.
Excellent presentation, very comprehensive clinical data you presented with very high impact publications. One question I have, this relates to your contributions, which are monumental, as well as radiation therapy in general. As a clinician, do you draw samples of blood from your patients during the therapeutic regimen, whether it is 10 days, 2 months, or whatever that is, and send those samples for analyzing IL-10 or do RT-PCR to find out if there is a substantial accumulation of pro-tumor M2 macrophage? By the way, IL-10 production is directly related to enhancement of pro-tumor M2 macrophage. This question is relevant because it's very well known that chemotherapy and radiation therapy, they do their jobs, which is reducing the tumor burden. But at the same time, they do contribute, to the enhancement of these pro-tumor signals. And if that is left unattended, it basically, defeats the very purpose. Yeah. So,
I understand, and that can be an area of interest where it can be done, but as a clinician, I think we don't go for into that details, but it can be definitely an area of interest and all this thing. We can think of it.
Okay. So the last question is from Dr. Pandey.
Yeah, very nice data. I got one more radiobiology related question related to our query, you can say. Like you have shown one place that we are using some beta emitter, alpha emitter together for studying the efficacy. So whether, see, beta emitters may cause some radio adaptation in radiobiology, what we say, and alpha emitters are likely to cause very less effect. So is there some studies where you, when you combine these two types of radiations like beta, especially for soft beta, and then the alpha, whether you find more sort of less response or radio adaptation for the response?
Okay. People are doing a lot of trials, and there are trials which are going on where alpha as well as beta both are being used in a patient, and probably they'll give us a result or outcome in a time to come. But definitely beta as well as alpha emitter, if they can be combined, it would be really good for the patient because it can tackle of the macro as well as the micro metastasis. So that is what I feel.
Yeah. Thank you, Dr. Sood, for giving a very vivid details of what you did as a clinician. Can we use this lutetium, the short-lived isotope, for seeding instead of attaching to a ligand and then approaching the site?
Lutetium, okay, sorry, what I understand, lutetium 177 has a half-life of 6.7 days. So it's not a short-lived,
but 179 and other options are also there for the isotopes of lutetium having short-lived isotope that can be seeded, and that can also help in
I'll not be,
okay.
I'm not the right person to answer
so
it.
Thank you very much.
Thank you.
Thank you very much for wonderful talk.
Thank you.
So I invite Dr. Ashish Soni for his talk, and he'll be talking about how telejoparib exerts robust radio sensitization through differential regulation of distinct alternative and journey pathways. So a very brief overview about his credentials are shown. So for the paucity of time, I will not read, and he's well-known researcher. So I invite you for your talk, please.
Well, thank you so much for the kind introduction. First, I would like to thank the organizers for giving me the great opportunity to present our work here. And I realized by looking at the first talk that I have to switch my gears because I will come back to the basic research and I will take you back to the field of DNA double-strand break repair, what George started yesterday, the plenary talk actually. And I will tell you the story that how we come to the bridge of basic research to bringing a compound at least to the level that we can check it now for the clinical efficacy. So as title already said by the respected chairperson, we saw that talazoparib exert a robust radiosensitization through differential regulation of distinct alternative and joining pathways. I will start with the slide. George showed it in very detail yesterday. I will not say so much about that, but I would like to emphasize that two main repair pathways are in the mammalian cells. Classical non-homologous end joining is the main pathway which is resection independent. And when the cells decide to resect, the CNHEJ is by nature excluded. Now, homologous recombination comes into play on those resected ends, and this is the error-free pathway only mammalian cells have. When the two main repair pathways somehow are compromised, then cells opt for the alternative end joining, and I will say today the PART1 dependent alternative end joining, but recently it's also have been shown that Pol theta has been an integral part of this kind of end joining. As George said yesterday, we have worked for quite a long to characterize this alternative end joining pathway, and what are the roles of PARP1 ligase 3 in the DNA double-strand break repair, because it is a well-known protein for the single-strand break repair, but you would appreciate that we found that its at the core of also double strand break repair, but on the cost of many errors which are made in the genome in the terms of translocations. Now, the PARP1 is a very versatile protein, and it is involved in many processes, as I said, and also different pathways. It's a enzyme which does a post-translational modification to various proteins in these processes, which controls the DDR signaling. And with this, what we were interested to look for that how the PARP1 is involved in alternative end joining and causing the ionizing radiation-induced translocations, and how the double strand breaks are repaired here. So first, how the PARP inhibitors comes into so much into the, sorry. I must go slowly. So these are two very important publications, came back to back in Nature in 2005, which bring PARP inhibitors to the forefront to treat the BRCA deficient cancers with the concept of the synthetic lethality. And many clinical PARP inhibitors which are shown here, like olaparib is very well used in the clinic. There is a very recent PARP inhibitor, talazoparib, which I'm going to tell you in my story today, and they are very promising, but as Nils very nicely said today, that there is a problem of the resistance to the therapies, which comes apparent very quickly, and we have to find the combined treatment modalities to overcome this kind of resistance. So we started actually looking at a very basic question. As I say, we are basic researchers, we do DNA repair. So what we asked, when we irradiate the cells, what happens to the genome? And if you see here, I have wild type cells and the mutants for different pathways, classical non-homologous end joining, homologous recombination, and the double mutant. When we see at 4 hours in a MAF cell after 1 gray irradiation, you would see that there is increased translocations when we don't have the pathways. And when both main pathways are not there, then we see a manifold increase in these translocations. Who is causing them? As we proposed the alternative end joining, we thought, well, let's inhibit the PARP and see. So we use PJ34. It's a second generation PARP inhibitor, and we saw that there is a significant reduction to those translocations. They are not yet gone completely, but we see a clear effect there. So alternative end joining plays a very important role here. Now, when we see for the radiosensitivity, you will see that there is a very moderate radiosensitization, and this is actually have been reported for other PARP inhibitors also, which are in clinic. And that somehow underestimate the potential of PARP inhibitors to use as a radiosensitizer. Now, this was 2014. 2015, there was the talazoparib, still not available in the market, but we got it through NCI, and our purpose was to see whether we can get rid of all of these translocations, because this inhibitor could not be the best inhibitor we could get. And to our surprise, we got manifold more translocations. I went to George and I said, George, I saw this, and I was going to Icrr to present my data there, and he said, oh, that's good. Have you repeated that? I said 3 times. He said, okay, let's think about that. You do not present this there because this doesn't actually not in the line what we are thinking, but let's check the radiosensitivity of those cells. And there was a master's student at that time, so we give her this project that check that. And this is what she did. Chinese hamster cell, you will again say, why Chinese hamster cells? Because we have many important mutants there, and as we have the basic research questions, we could address them with the Chinese hamster cells very well. So we first use them, and you will appreciate that in comparison of ionizing radiation alone, or the other PARP inhibitor like PJ34 or olaparib, BMN 673 give a very strong radiosensitization. We saw it even at the 10 nanomolar concentration, which saturates at 50 nanomolar. So we decided to use 50 nanomolar, which is a very good concentration, which is also clinically achievable. Now, we asked that why this inhibitor is so different. So maybe it is, there is some difference in the PARylation inhibition. So we used those inhibitor and looked for the PARylation after H2O2 treatment. And as you can see, that PARylation is completely inhibited. So this cannot be due to the catalytic, just difference in the catalytic inhibition of those proteins, but there is something more than that. And as we are the DDR repair repair lab, we wanted to check in detail what are the differences, which I'm going to show you in the next few slides, what we have found and what this talazoparib has unique in its mechanism of the radiosensitization. Now for clinicians, it's very important to use the human cells or cancer cells. So we were also a part of this consortium, which was the sarcoma consortium with the NCI. So we used the cells, rhabdoid tumor cells and the sarcoma cells, and you can see that as in CHO cells, we can very well radiosensitize them. Now here there are two protocols. I treat the cells only for 1 hour with BMN, talazoparib, or we keep the drug for the whole time, for example, and you see that there is a radiosensitization there. PJ34 doesn't do any radiosensitization in these cells. So bottom line is that even after 1 hour of treatment, if you see, we could get maximum radiosensitization in many cells, if not all. And I will show you also later a few examples which suggest a flexibility in the treatment you can have in the clinic. Now, also we wanted to check the normal cells, so we took two, human fibroblast cells and epithelial normal cells, and to our surprise, there was no radiosensitization at any concentration we used of the talazoparib. So this is another very good character of an ideal radiosensitizer, that we save the normal cells and only sensitize the tumor cells. So what is happening at the DNA level? Now I will show you this in few slides. So talazoparib, when we treat the cells, this is another PARP inhibitor, AG14361, and you would appreciate that this is the control, and 2 gray after 8 hours, almost all foci are gone, means double strand break repair is done. But almost majority of the breaks remains unrepaired, so gamma H2X is still there. So we see that BMN inhibits the repair. Now double strand break repair, now we know 4 pathways. So we still don't know which pathways are inhibited. We looked for the transition like double strand break and resection, because this will exclude the NHEJ and go the pathway switch to the homologous recombination or alternative end joining. In comparison of the control, you will see BMN treated cells show manifold more double strand breaks which get resected, which means that there is a switch to the resection dependent pathways. Could it be homologous recombination? That was our next question, and we opt RAD51 as a surrogate marker for this, and you see here that there are more RAD51 foci. But we don't see in reporter assay any change, so homologous recombination is not changed. At pulse field, we could see that the BMN is like DNA PK inhibition, and the combination gives an additional suppression of the repair, so both classical non-homologous end joining and alternative end joining are suppressed. I don't go in the detail because we don't have the time for that, but we know this, that this assay mainly measures classical non-homologous end joining and alternative end joining. And lastly, this was the translocation slide which I showed you in comparison of other PARP inhibitor. We see manifold increase in these translocations, which gives this tremendous radiosensitization in these cells. Now, we checked many cells. Some were very good responders, but some were, many of them are moderate. And how we can solve this? Because they are not, here talazoparib was not efficacious. You can see as this is the DMF at 10%. So we looked how we can, what is going on there, because it looks like that there is still a subset of alternative end joining which we mark, and which could be Paul theta dependent, because this is another component of this pathway. So one of our students, he checked this in the human and the hamster cells, and in the responders, talazoparib gives no additional radiosensitization, but you will appreciate that those moderate the sensitive cell lines give very robust radiosensitization when we combine talazoparib with the Paul theta inhibitor. So still we had room to suppress other pathways or parallel pathways of alternative end joining, and you will see that we can bring the moderate responders to the level of the responders, which we have, this paper we have just published recently in the Green Journal. And these are the mechanisms. I will not take so much time here, but we see that the resection is very important player, as I showed you also in the beginning. And if we suppress the resection by downregulating CTIP, which is the main protein, we see that this sensitization in POLQ deficient cells is no more there. Additionally, the translocations, which I show you before also, the talazoparib increase the translocations manifold. They are first resection dependent, because in the CTIP depleted cells we don't see that increase, and also in the Paul theta. They are Paul theta dependent cells, Paul theta dependent in the control of DSP and resection. So with this I come to the conclusion. So at least I hope I convince you that we have a compound which has many properties to be an ideal radiosensitizer in the clinic, and which can be used at low clinically doses and in the flexible treatment time regime. Talazoparib specifically radiosensitizes tumor cells, and combining talazoparib with Paul theta inhibitor gives us the window to further radiosensitize those cells which are resistant to the therapy. And overall, all this can be done independent of the HR-ness of the tumors. So we are not dependent whether they are BRCA deficient or HR deficient. We can radiosensitize a wide spectrum of tumors. At least that is the promise. And we want to take it to the preclinical models, also to the 3D culture first, and then of course it has a potential to be implicated in the clinic. With this, I would like to thank George. Over the years, he helped me. He's my mentor. Professor Martin Stuschke. This is the student Shishi Lin, who just submitted his thesis, and he was involved in the Paul theta project, and all our funding agencies, and all of you for your kind attention. Thank you so much.
Thank you, Dr. Soni, for being well in time. We are running quite a bit late, so we'll take only one or two questions at the most, and be very specific and short, please.
Thank you, Dr. Ashish, for your nice talk. So I have just one question. As you know that after DNA breaks, the cell has two choices, whether it will go to HR pathway or NHEJ pathway. So, and it depends on the intensity of the damage and the genetic setup of these cells. So why do you think that, this talazoparib, it predominantly inhibits NHEJ pathway?
Inhibit NHEJ pathway?
NHEJ pathway?
That is actually what we have seen, that NHEJ is first NHEJ and then alternative end joining. Although we see hyperrecombinogenic phenotype, but this doesn't reflect in the survival. You see that if more homologous recombination, we will expect radioresistance, but this is not what we see more radiosensitization.
And do you have any data about the expression levels of these HR candidate genes after treatment of PARP inhibitors?
Which kind of genes, for example?
For
DDR genes, you say.
DDR.
We have checked some of them, and Shishi, he's still maybe checking some of them, like the expression of PARP1, Paul theta for sure, and we will have this result soon. Now we are really narrowing it down, what is happening.
Thank you.
Thank you very much.
The last question, please.
A very brief question. Do they have a class action? These are all PARP inhibitors. Do they all behave the same way, or this drug is unique and things like that?
Actually, this is what in the beginning I showed you.
Sorry, I was not there.
They inhibit the PARP completely. So the catalytic inhibition is complete.
So this
what is happening is, at least we say that, and it is happening actually. We see that this is happening on the double strand break itself.
I get that. So class action is, say for some of the antipsychotics, they don't have a class actions. So do you think PARP inhibitors as a group of drugs will behave similarly or differently?
Sorry, I didn't get your question because please.
Yeah, okay, sorry. Now, say for some of the antipsychotics, they don't behave the same way, though they are antipsychotics. These are PARP inhibitors. There are few PARP inhibitors. Do they all behave the same way as this particular drug is behaving? And then maybe, what is true for this would be true for some other PARP inhibitor. That's what I was asking.
I don't know. I can talk to you later also, but at least you see that we checked a battery of PARP inhibitors, which only olaparib gives us some intermediate effects. So they are not same for sure. They have differences, but definitely different kind of tumors should be screened for their response.
We can discuss over tea.
Thank you.
So the next speaker, I will invite Professor Utpal Ghosh. He's from University of Kalyani, and he's going to talk the combined effect of high LET carbon ion with this PARP inhibitor, olaparib, for treating highly aggressive kind of human non-small lung cancer cells, which may be considered as a promising cancer therapy. Professor Ghosh, please.
Thank you very much. Thank you very much for your kind words. So myself, Utpal Ghosh. First of all, I would like to thank to the organizers for giving me opportunity to share my thoughts and works over here. So today's my talk is high LET carbon ion combined with PARP inhibitor olaparib. It could be a promising approach for cancer therapy in highly aggressive human non-small lung cancer cells. So the content of my talk is, first I will give a very brief introduction about the carbon ion radiotherapy. I think most of this part has been covered in earlier speaker covered these things. And also I will introduce what is PARP biology. And then objective of my talk is combined effects of carbon ion and olaparib, PARP inhibitor, focusing on DNA damage and repair pathways. Little bit I will show few slides about the apoptosis induction, and then I will show signaling pathways of reduction of metastasis. So carbon ion radiotherapy, it has its own inherent advantages over conventional radiotherapies because it has an excellent dose localization. As you know that radiation causes cancer and radiation kills cancer, so targeting matters. And it has a very effective, it is very effective against hypoxic tumors and certain type of human cancer, those are inoperable, like NSCLC. And regarding PARP biology, I think Dr. Ashish describes, this is, so this is the target of the PARP, target proteins of the PARP. PARP basically poly-ADP riboses, and each ADP ribose moiety carries a negative charge. So it modifies the surface protein of the target, surface protein of this target, and thereby it changes the function of the protein. And there is another group of protein which cleaves this polymer and reverts back to d-ADP ribosylated form. So basically there is an equilibrium between poly-ADP ribosylation and d-ADP ribosylation. And it has a very close relation with radiotherapy because after the DNA damage, this poly-ATP ribosylation reaction shoots up, it's accelerated, and after that it decays. And there are known PARP inhibitors which are in clinics. It is long been known as the chemosensitizer and radiosensitizer, as evident from earlier lecture also. But it has some other functions also, as evident from recent publications, other than this radiosensitization effect interfering the repair pathways. It can modulate the gene expression of several oncogenes, and also including some transcription factors. So it is very, sorry, how do I do this? So it's a key player in cell cycle regulation, transcription, apoptosis, and also metastasis. I will show here few slides of the how it regulates in reduction of metastasis. And olaparib, as you know, that it's effective against BRCA1 2 mutant breast and ovarian cancer cells. So why combined therapy? Actually, my objective is to see the combined effect, because if you combine these modes of therapy, then maybe you can reduce the side effects, and also you can target the multiple pathways of cancer. So my objective is this DNA repair, HR and NHEJ pathway. And I will show a few slides about the metastasis inhibition. So this is I am not at all doing any therapy. I am doing in this work in the cell culture model. I use this P53 wild type F549 cells and P53 null H1299 cell, and we measured DNA repair activities, investigated DNA damage induced apoptosis, and also some markers of EMT signaling pathway that shows it also acts in the reduction of metastasis. So if you look into this survival curve, it's very interesting. P53 null cell is so solder, that means it is to some extent resistant to carbon ion radiation. But if you combine with olaparib along with this carbon ion, it is straight, becomes very sensitive. So take home message that, as you know, that the almost 50% cancers harbours P53 mutation, and those mutations having the inactive P53 could be effective in treating carbon ion with olaparib. And if you knock down PARP1 gene, especially, it sensitizes. It is well known. I am not going into the detail, but radiosensitization is higher if you compare carbon ion with gamma radiation. And if you compare this, we also checked this radiosensitization in the other type of cells, where we found that if you see the radiosensitization X-rays compared with the proton beam, compared with this with the carbon beam, so carbon beam it sensitizes more. And for each type of radiation, if you combine with olaparib, you see the result, it sensitizes. And most of the sensitization, it is found in the carbon ion. Now we checked that repair pathways after carbon ion exposure. We checked the poly-ATP ribosylation with time kinetics, and I have seen that immediately after 1 hour, the poly-ATP ribosylation shoots up, and it creates DNA breaks. And if you allow to allow the grow to for some time, then some of the damages gets repaired. But if you treat with olaparib, in presence of olaparib, if you treat the cells olaparib before the treatment of carbon ion, it drastically reduces the repair potential. And if you look into the expression of these HR candidate genes, you see here RAD50, RAD51, MRE11, those are the candidate genes for HR pathway. Immediately after exposure of carbonion, there is a trend of increase of this protein expression, and it is obvious that after damage, cell try to repair it, so expression goes up. But if you pre-treat the cells with olaparib, you see a significant reduction of the expression, and that is very important from the basic science point of view. PARP by its poly-ADP ribosylation reaction can modulate the expression of proteins. So that is the key factor for using this olaparib, and that's why it is giving versatile effect, more effect in cancer therapy. And in 24 hours, trend is that it is reducing, and when you combine with this olaparib, so it reduces the expression of these NHEJ proteins also, Ku70, Ku80, here's the part in the NHEJ. So, so take home message is that if you inhibit PARP, it actually reduces the expression of the candidate genes in repair pathway. And it is also interesting that as Professor Nagaraj was asking, it is not so simple. When you combine olaparib with carbonion, it drastically reduces both HR and NHEJ pathway, as evident from my work. But if you combine with the other chemotherapeutic agent, the mechanism is different. If you see here that only a P38 inhibitor, it reduces HR pathway, but doesn't have any effect on NHEJ. But when combined with this PARP inhibitor, it reduces the error-free pathway and induces the error-prone pathway, NHEJ. So the mechanism is different. So PARP inhibitors, it is very effective with the radiotherapy as well as the other chemotherapeutic drugs, but mechanisms are different. Now, I have investigated the total apoptotic pathway. I am not going into detail, but if you treat the cells alone with this PARP inhibitor, or if you knock down PARP1 gene, it induces apoptosis, and if you treat this carbonion, so induction of apoptosis is higher in case of knockdown cell, and similar kind of results obtained when you treat these cells with the olaparib. So, but what I would like to focus here is that a role of PARP in metastasis. If you knock down PARP1 gene, there is a little bit reduction of metastasis, matrix metalloproteinase 2, 3, 2 and 9 going down, and if you irradiate these cells with carbonion, so drastically reduce the activity as well as its expression. So, not only PARP1 interacts by its poly-ADP ribosylation reaction. In fact, this is interest of basic research. If you look into the structure of this PARP1 gene, it has a catalytic domain by which it can poly-ADP ribosylate. There is a domain which is called the protein-protein interaction domain, and it can interact with the so many proteins. So I am not showing those data here, and possibly that's why the other effects we observed when we knock down PARP1 gene than when we inhibit its catalytic activity. If you compare the cell migration or MMP's activity with P53 wild type and P53 null cells, it is different, and when you combine this with olaparib, the reduction is significant for P53 null cells also. So this is very interesting result. It indicates that
P53 mutation, which is in some cancer which is becoming very aggressive and difficult to control, could be effectively controlled by carbonion combined with olaparib. And if you look into the whole signaling pathways, EGFR, AKT, P38, all these protein inhibitors are individually applied for cancer therapy. And if you look this data, that with olaparib treatment and also the SI knocking down of PARP1 gene, there is a difference of expression, as well as the most important thing is that you see here phosphorylation is being changed of this protein if you treat the cells with the PARP inhibitor. So that means, that indicates that olaparib, it inhibits the poly-ADP ribosylation reaction of PARP1. It affects the phosphorylation of different key proteins in DDR pathways. And so, but if you look into the expression pattern in these two cells, they are different. But ultimately, if you look that combining PARP1 inhibitor with this carbonion, it drastically reduces the metastatic potential by this pathway. So this is the whole pathway. So MMP29, N-catherine, vimentin, claudin, aniline, and this red one shows the effect only if you inhibit the PARP1, and the green one is the effect only carbonion, and the blue one is the combined effect. As you see from these signaling pathways, if you combine this, it sensitizes more the cancer cells. So this is the key findings. So combining carbonion with the PARP inhibitor, it not only interferes the DNA repair pathways, it also reduces the metastasis, and the pathway which it interacts for the basic science studies, PARP1 interacts with so many proteins. So these proteins are individually, inhibitors of these proteins are used as the cancer therapy. So in one shot, if you inhibit the PARP1, it in other way you inhibit the whole pathway, so that drastically reduces the metastatic potential of the cancer cells. So I must thank to IUSC personnel, where I use this infrastructural facility for irradiation of carbonion, and these are my students and collaborators, and these are the funding agencies. So thank you very much for your attention.
So thank you, Dr. Utpal, for a very lucid illustration of the synergistic effect of carbonion as well as the PARP inhibitors. But over to the audience for a short question, if any. Just one short question.
Yes, Dr. Fantastic presentations. So where you use the carbon source?
IUSC, Inter-University Accelerator Center, New Delhi.
So they have carbons. And
Yes.
what is how is it set up? Is it fluence base or dose base?
You can change the dose from 0.5 gray to 4 gray, and if you change the fluence, dose also will change, because if you look into the formula, that dose, it depends on the fluence also.
Because you mentioned that carbon is better than the protons.
Yes.
So
yes.
and what's the protons is same center as also as a proton facility?
No. Proton facility I have used in Genil, France.
So how can you compare one in India, one in Galin, the two the same?
The doses are same. If you compare the dose of the particles, you don't have any problem. But some sort of, if you think about the photons and with the particles, but there is a normalization. So we did that.
Okay. I usually prefer to have
because particles
okay.
nature and the wave nature, they have the different way of doing energy deposition. So from the energy deposition concept, you can calculate the dose.
Okay, thank you.
So thank you very much. Maybe we can talk more about it during the tea break. So over to Dr. Richa Chauhan for the next talk. She is a senior consultant radiation oncologist at the Mavi Cancer Sansthan, and she has her research domain as radiation and cancer biology, biomarkers and health economics. She has 66 publications to her credit, and we would want her to deliberate on proteomic insights in the chemoradiation therapy resistant cervical cancer patients, for which she also has a patent. Thank you.
Thank you, ma'am. A very good morning to all. So in the next 10 to 15 minutes, I'd like to discuss my work and throw some proteomic insights in the chemoradiation therapy resistant cervical cancer patients. So just after a very brief introduction, I'll discuss the objectives, the method, the results which I got, the concluding, the future prospectus, and of course the most important, we do acknowledge the people who have helped you. So we all know that cervical cancer is still a major problem. It is one of the most prevalent gynecological malignancies in women, contributing to close to 6 lakh cases. And if we talk about the incidence, mortality and 5-year prevalence, this is the highest in Asia. If we talk about the cervical cancer burden in India, it is still the second most common cancer, and 1 in every 5, that is 20% cases of cervical cancer, and 1 in every 4, that 23% is because of cervical cancer is in India. The point I want to highlight is that cervical cancer still, in spite of the availability of the HPV vaccine and a very effective screening method, it still remains a major problem for our patients. Okay, so we all know the stages. So I'm basically confined about the locally advanced cervical cancer stages, in which chemoradiation is the standard treatment. Why? Because we have the POX study by Nandkumar et al. That reported that even today, close to 80% of cervical cancer patients in India, they present in a locally advanced stage, that is from stage 2B to 4A. If we include the new staging, even 1B3 is included in a locally advanced stage. And cisplatin-based concurrent chemoradiation has been recognized ever since the announcement by NCI in 1999. This has remained as the standard treatment for these patients. And despite being a relatively radiosensitive tumor, we have the advantage of giving brachytherapy boost doses to the cervical cancer. About 30 to 40% of these patients fail to show complete response to chemoradiation, and they progress with a poor outcome. So this is what happens. This is just a representation. We usually give them 5 weeks of external beam radiotherapy along with weekly cisplatin, which is followed by 3 to 4 fractions of brachytherapy. And then after 4 to 6 weeks, we assess the patient for response. And then only we can differentiate whether our patient has responded or non-responded. So this takes almost 3 to 4 months. An improved understanding of the underlying genetic and proteomic alterations involved in the process of carcinogenesis and those that mediate the resistance and relapse would offer significant opportunities in improving outcome across the spectrum of cervical cancer patients. So we thought that the heterogeneous response of patient towards concurrent chemoradiation demands profiling of each patient. Hence, biomarkers for selecting the right cohort would be a strong tool to personalize treatment. See, this is what is happening now. We are treating the patient, and then only after treatment we may know that the patient are responding or not responding. So we thought if we can have a pre-treatment biomarker from the tissue biopsy, which can be scored and which can be validated simply by IHC, because all our patients, they cannot go for NGS or systemic omic profiling, and we can know who are the patient going to respond, who are not going to respond, and treat the resistant patient with an alternative therapy. So this is how we aim to improve the results. So with this concept, and further therapy resistance occur due to multifactorial events and targeting the key molecules. We also plan to study the pathway, the phosphoproteomic involved in treatment resistance could combat treatment failure so that we could get some target molecule for those patients who are resistant. So we profiled both the global proteomic and the phosphoproteomic landscape of the concurrent chemoradiotherapy resistant cervical cancers. So these are the objectives. We wanted to identify the differentially expressed proteins in concurrent chemoradiation resistant and sensitive cohort of patients. We wanted to identify aberrantly activated kinase and also to validate the differentially expressed proteins and activated kinase in the different cohorts. So, but today I'll discuss only about the global proteome of the concurrent chemoradiation resistant and sensitive cohort patients. The material, we collected the tissue biopsy sample from patients before we started the treatment, and the patients were treated with the standard protocol, and clinical response were evaluated as per the RECIST 1.1 criteria, which we use in our clinical practice. Patients were either complete responders, any patient with a partial response, a progressive or stable disease were grouped together as a non-responders, so that we have only 2 cohorts. And this is the simple workflow. After taking the biopsy, we did a protein extraction, the simple pathways TMT labeling, and then that was subjective to a mass spectroscopic study, and data was analyzed. So what we found in that data, the spectroscopy data processed and searched against data, we used the Sequest HT and the mascot algorithm, and we could identify around more than 8,000 proteins, and out of this, more than 7,000 proteins were quantified across all samples, and the PC analysis showed clustering of the 5 samples in each cohort, and 2 cohorts separated as 2 distinct clusters. So these are the responders and these are the non-responder cohorts. So this is a volcano plot of these, 7,138 proteins who were quantified. So out of them, 73 proteins were significantly expressed between the cohorts. 11 proteins were upregulated. These 11 proteins were upregulated in the non-responders, and 13 were downregulated in the non-responders. So we could find some significant finding. Further, this is the list of top 10 dysregulated proteins which was identified in our study by quantitative proteomics. We can see serpin B7, syntaxin 3, LBP, amylin. These are the list, and the most, these have been arranged to their significant value. And this is a heat map which of the top 5 upregulated proteins in the non-responders. We tried to look for them and search if you can do with something with them. And these are the pathways which is soon to be enriched in the chemoradiation resistant patients. So the major pathway was the sister chromatid cohesion, which was involved in chromosome segregation and homologous recombination of DNA double-stranded repair. So this was the major pathway which we found was associated with the proteins that were upregulated in the non-responders. So next we plan to do a survival analysis. We got few proteins which were significant and they were upregulated in the non-responders, but whether do they have any effect on the survival of the patient? And to determine this prognostic significance of the dysregulated proteins, we assessed their correlation with the survival of cervical cancer patient using the C bioportal, and we generated survival plots for these top 5 regulated. I'm showing you the survival plot for the 2 top regulated protein. The median, you can see the median overall survival, the difference in the survival when there is an upregulation of these proteins and when they are not upregulated. So the median overall survival of patient with an altered expression of syntaxin 3 was 40 months, and in our data we also observed high expression of syntaxin in non-responder cohort. So further we worked on syntaxin 3 because we looked into the literature, we did a data search, and we found that yes, syntaxin 3 has also been previously reported in, in some cancers like breast cancer, prostate cancer, and high expression of syntaxin 3 in breast cancer was correlated. It has been already correlated with poor overall and disease-free survival, and it reduces the PTEN stability to activate the mTOR pathways. So we found some reference, so we thought we should move ahead with syntaxin 3. So it was further, we wanted to validate in other cohorts and groups of patients and confirm it on the IHC. So based on our data, we thought to determine the differential expression in larger cohorts, and then we collected the FFPE tissue samples of non-responders and responder of both stage 2B and stage 3B, and in concordance with the proteomic data, we observed high expression of syntaxin 3 in the non-responders. So this is the result, the staining. You can see a high expression, low expression in the responders, and this is, so what we found significant was that around 83% of the non-responders showed medium to high expression of STX3 even on IHC. So coming to the results, we present the first comprehensive proteomic profile of treatment-resistant cervical cancer patients, providing a valuable foundation for identifying potential biomarkers. We quantified more than 7,000 proteins across the responders and non-responders, and 73 proteins were significantly enriched in the non-responders. The proteins serpin B7, syntaxin 3, LBP, amylin 2, and NQO2 were top 5 highly expressed proteins. Out of this, a high expression of STX3 was confirmed by IHC, suggesting the predictive potential of the protein. So we can conclude by saying that syntaxin 3 can be used in clinics as a predictive biomarker for selecting the patients who will or who will not respond to a concurrent standard radiotherapy. And phosphoproteomic analysis can further reveal distinct phosphorylation events and signaling pathways involved in treatment resistance, and targeting the activated kinase could combat treatment resistance in cervical cancer patients. So further perspectives, we need to do a comprehensive validation of biomarkers on a larger cohort, ensuring robustness, and then we need to translate the predictive biomarker from lab research to clinical standard for guiding the chemoradiation resistant patient. And we also plan to develop animal model to investigate the resistance mechanism, go back and study the resistance mechanism. So as a result of this study, we applied for the syntaxin 3 as a potential biomarker, and luckily we got the patent from Government of India. And we have also completed the phosphoproteomic study in which SMAC1 and casein kinase inhibitors, they have been found to be the main pathway, and we got a drug, silmetasertib, CK4945, and that has shown very good results, and we have published, the paper has been accepted, not yet published. We did a phosphoproteomic guides low-dose drug combination of cisplatin and silmetasertib against concurrent chemoradiation resistant cervical cancer. So, and before I conclude, I must acknowledge Dr. Irin. She was a PhD student from Institute of Bioinformatics. Dr. Kumar, Chief Scientific Officer, now shifted to Datar Cancer Genetics. All the faculties and residents of my department at Mahavir Cancer Sansthan, my HOD, Dr. Vinta Trivedi, and our directors for the administrative supports. So thank you all.
Thank you, Dr. Richa. One question from the floor. So if not, then we can, before moving to the next, I would just like to ask, have you validated, as you also said that it is multifactorial, the resistance is multifactorial, so, or the response is multifactorial. So did you validate with your demographic profile of the patients how long the treatment time was, what was the anemia relation with the patients and everything?
Ma'am, to remove all these bias, we when we selected the sample, we took only all those samples who were either stage 2B, we evaluated them separately, or stage B, patient who are having, similar hematological profile, renal profile, completed the planned treatment in the planned protocol. So we tried to remove as much bias as possible in the, the treatment factors and the demographic factor. We were only looking at the molecular factors.
Thank you very much, Dr. Richa. So now moving to the last, we have one question. Yes, one question, please.
Thank you, Dr. Richa. Very nice presentation. I have a question, like for the 5 up-regulated proteins, you did a confirmation, a validation kind of a study?
Yeah, we did that. No?
Immunohistochemistry only?
Yeah, actually we wanted to move forward with the top upregulated proteins, and first we had a search in the literature so that we can find some reference for moving further. So we found good reference for syntaxin 3, and then when we looked for antibodies, we had an antibody which was available, so we could practically do IHC with syntaxin 3. Because cervical cancer is basically a disease of poor patients. So all our patients, they cannot go for an omic profiling. So we wanted something which could be easily done in the lab, and IHC was, we thought, the best way out.
Coming to this, are you not planning to go for a panel of proteins to be more confirmed in this? Like a single protein biomarker, I think a panel is more.
Yeah, actually we have also done the whole genomic sequencing of these cohorts of patients, and we are still integrating all the data. So we plan to have a gene panel, a targeted gene panel for these patients. We have got some novel, EGFR amplification and all that, but we are still in the process of compiling those data. So we are planning to get a targeted gene panel for these cohort of patients.
And from proteomic point of view, are you planning to go for a targeted proteomics validation, because this is the most modern technologies where PRM, MRM people are using, that is for multiple reaction monitoring,
Yeah, but practically we are more focused on finding a drug, so we are more focused on working on the phosphoproteomic pathway.
Discuss this during
and
the tea break. I think we're short of time. So for the next, I would like to call upon Dr. Sudapa Mukherjee. She is a senior scientific officer at the CNCI Calcutta, and she will be delivering her talk on the ROS-driven HIF-1 alpha Icrr signaling axis triggers acquirement of radioresistance in cervical cancer. Her credentials are given. Over to Dr. Sudapa. Thank you.
Thank you. So, good afternoon, delegates. It's my pleasure to be a part of this prestigious meet of Icrr. So, the title of my presentation is ROS-driven HIF-1 alpha ORCA signaling axis triggers acquirement of radioresistance in cervical cancer. I am not going into the details of cervical cancer, its therapeutic regimen, as it has been already described by Dr. Richa. So, when cells are exposed to chronic doses of radiation, we all know cells adapt itself in such a way so that they can escape radiotherapeutic challenges, and ultimately these cells fuel resistance and finally metastasize to distant site and instigate disease recurrence. So we all radiobiologists know that the success of radiotherapy depends on the phase of the cell cycle, phase at which radiotherapeutic challenges are given. So considering this, G2M is the most radiosensitive phase, so escaping this G2M phase is the strategy for rendering radioresistance. With this, considering the major challenge in the success of radiotherapy, we were focusing on some protein or molecule that is responsible for this facilitating G2M phase. So in our laboratory, we are working with one mitotic serine threonine kinase, that is aurora kinase A or ORCA, that facilitates G2M progression. It is a centrosomal protein. It helps in spindle assembly and all the mitotic event and G2M progression. Not only that, already we have observed that this particular protein is responsible for rendering chemoresistance in cancer. And TCGA database also shows that it is associated with high expression is associated with poor survival. So based on these preludes, the question came into our mind whether at all this particular kinase, ORCA or aurora kinase A, is at all dysregulated in the scenario of radioresistance. For that, we took cervical cancer biopsy samples from patients. We took almost 55 patients who were visiting our clinic, CNCI OPD clinic. We took biopsy samples, and first we checked the expression of ORCA in these samples. So based on ORCA scoring, we categorized patients into two categories. One is high ORCA with high score, and another batch with low ORCA with lower score. So these are the stages that we taken into consideration, and not only that, these are some other clinical pathological parameters that we taken care of. So based on ORCA scoring, we further extended our work to check the ORCA profile by flow cytometric analysis. So in all these two groups, group 1 and group 2, we checked ORCA expressional status, and here you can see that in group 1, ORCA expression is much higher compared to group 2. And this is the ORCA positivity. Not only that, we took the repeat biopsy as well. So after post-radiated samples we took, and again we checked the ORCA expression, where also we found that upon radiation, ORCA expression, even it is higher upon radiation. As I have mentioned that we performed IHC, and these are the IHC scores as I have discussed. So the possible conviction that came into our mind whether there is any altered, because radiation generates ROS and alters the redox imbalance. So whether redox imbalance has any impact on this expression of this particular mitotic regulator. So for that, first we checked the ROS level in these two batches, and here it is clear that group 1, which are high ORCA group, their basal level of ROS is quite high, and which is even higher upon radiation, compared to group 2, where basal level of ROS is lower compared to group 1. Not only that, we checked the cell cycle phase specific ROS as well. For that, we first checked the cell cycle distribution pattern, and here you can see in group 1 with high ORCA, there are hardly any G2M population even before or after radiation, whereas in group 2 with low ORCA, here G2M population increases upon radiation. Not only that, when we looked for cell cycle phase specific ROS, then we found that in group 1, S phase ROS is high compared to group 2, and which is even higher upon radiation. But in group 2, which is low ORCA group, their G2M ROS significantly increased upon radiation. So, considering mitochondria as the chief source of ROS, we checked for mitochondrial ROS, and some literature studies have already shown that some mitochondrial localization of ORCA. So based on those literature studies, we first checked the mitochondrial ROS pattern in these two batches of patients, group 1 and group 2, and here you can see that in group 1, there is mitochondrial ROS level is higher compared to group 2. Not only that, we performed JC1 membrane potential assay, and here also in group 1 membrane potential red versus green ratio, you can see it is much higher compared to group 2. So it indicates that in group 1 patients with much higher mitochondrial ROS level, it is becoming, mitochondria membrane is becoming leaky. So it is expected that when group 1 having leaky mitochondria, so chances of cytochrome C release will be more. So with that notion, we checked the viability assay first, and we found that our assumption is not in alliance, because in group 1, even if the leaky mitochondria upon radiation, viable cells are less, more compared to group 2, and mitochondrial release of cytochrome C is less. No release has been observed in group 2, but it has been observed in, in group 1 with high ORCA, but in release has been observed in group 2. And regarding mitochondrial accumulation of ORCA, we found that yes, ORCA is being accumulated in group 1 patients compared to group 2. So far we have observed that two batches of patients, one with high ORCA and another with low ORCA, and mitochondrial ROS is more in high ORCA. In spite of leaky mitochondria in high ORCA group, cytochrome C is not being released. So what is actually going on in vitro setup for that? We don't know what will be the radiotherapeutic response pattern in these batches. So to validate our experiment, we performed in vitro setup. We developed one radio-resistant SHIHA-RR cell line from parental SHIHA. We took cervical squamous epithelial cell line because the number of patients we received all were of cervical squamous carcinoma. So we developed radio-resistant subline, and again we checked, and we found that in SHIHA-RR G2M population is not increased upon radiation. Not only that, ORCA phospho-ORCA expression was quite a high here, and as observed in case of patient population, here also we found that in SHIHA-RR, S phase ROS is higher compared to parental SHIHA, but in SHIHA, radiosensitive SHIHA, G2M population becomes higher upon radiation. Not only that, in SHIHA-RR there is a persistent elevation of ROS levels, and then we synchronized the cells, and here it is clear that when we synchronized S phase, then S phase ROS in SHIHA-RR is much higher compared to parental SHIHA. So whether this elevated ROS is at all facilitating mitochondrial entry, so in vitro setup also we found that in SHIHA-RR already there is a elevated ROS, mitochondrial ROS and membrane potential. And here also we didn't found any release of cytochrome C. And our immunoflow cytometry results also in allowance with our observation. Next, to further validate, we transfected ORCA gene in SHIHA, and here also we found similar results. So the question came into our mind whether there is any intermediate molecule, because we found high ROS, we found high ORCA. So is there any intermediate molecule, for which we checked HIF1 alpha, and we found co-expression of HIF1 alpha and ORCA in these two batches, particularly in group 1 you can see, and also in cell line as well. Not only that, we found that cheap assay showed that HIF1 alpha can bind to the 1 kb upstream of ORCA transcription start site. And when we inhibited HIF1 by ketamine, then you can see HIF1 is being inhibited side by side, ORCA is being inhibited. But when ORCA siRNA or LY3295, which is specific ORCA inhibitor, is used, then ORCA, yes, ORCA has been, number of ORCA positivity has been decreased, but not the HIF1 alpha. So based on these findings, we came to conclusion that probably ORCA is being upregulated through HIF1 alpha, and ROS is the mediator that triggers HIF1 alpha and thus ORCA. So what are the possibility to sensitize? So for that purpose, we used aspirin, which is already a known radiosensitizer, and it has moderate to low side effects. So first we checked the IC50 values, and we treated aspirin and checked the survival fraction in SHIHA and SHIHA-RR, and we found that SHIHA, which was initially not responding to radiation, was responding better in presence of aspirin. So what are the probable mechanism? Then we checked and we treated the cells with aspirin and radiation, and we found that pycnotic nuclei and hypernuclei and micronuclei and multiple nuclei, and DAPI-stent cells also showed DNA damages. So all these results shows that probably aspirin is inducing mitotic catastrophe. And not only that, aspirin could efficiently minimizes the S-phase ROS and also responded better against radiation. So we again reperformed the cells using under primary cultured condition, and again aspirin behaved in a better position. So in silico study we performed, and we found that there is an interaction between ORCA and cytochrome C, and probably that might be the reason that in spite of the leaky mitochondria, cytochrome C is not being released from those cells, and aspirin has been found to bind with this particular domain. And not only that, co-IP results also shows that yes, in presence of in absence of aspirin, there is no binding potential of cytochrome C is higher, but in presence of aspirin it reduces. So the overall message that I want to convey here is that during chronic irradiation there is a elevated ROS, particularly I must mention S-phase ROS, and that upregulates HIF1 alpha, and HIF1 alpha binds to the promoter region of ORCA and upregulates ORCA, and ORCA performs two functions. One is probably it is blocking, although I have to do some much more extensive studies in this regard, but it is blocking cytochrome C release, and it affects cell cycle progression. So I must acknowledge the director, our director CNCI, Chittaranjan National Cancer Institute, Department of Radiation Physics, Department of Gynecologic Oncology, CSIR for funding, and my lab members. And this is my team, and this work has been done by Shalini. This is her thesis work, and this is the institute where I work, and recently our institute has come up with a new campus at Newtown with all modern amenities and infrastructural facilities. And thank you for your patience hearing.
So, thank you, Dr. Sutapa. Very interesting work indeed. So we have time for one question. Yes.
Very interesting results. I got one question, like you are looking for mitochondria. So have you, first one is that HPV, since it is a cervical cancer. So what is status of HPV in these patients and whether they also affect the...
Status of? I didn't get it.
HPV.
HPV?
Yeah.
Yes.
That whether they have you evaluated whether they also affect your sensitivity of the cells and mitochondrial response?
Actually the sample size that I told you, 52, and I didn't check the HPV status, but it has been checked in our clinic, and they were, most of them, barring a few, most of them were HPV 16 positive.
That will be if you correlate.
Yes,
actually,
and the thing that I told that probably ORCA is going into the mitochondria, that here one gap is there. I know that I have to do further research how ORCA is entering into the mitochondria, because it is a centrosomal protein that we have to work on this, that part is also pending. How it is entering into the mitochondria, and it is preventing release of cytochrome C in spite of the fact that the membrane is leaky membrane. So that we found that there is an interaction between ORCA and cytochrome C. Apart from this, whether any other mechanisms are there, that we have to check.
Yeah.
These are pending.
So anyway, I know that in patients, most of the patients will have HPV positive, but at least in vitro level you can see this study. That will be basically, actually we got already HPV positive cell line and negative cell line. That will be very
That's why that's the reason I have chosen SHIHA, which is a HPV 16 positive cell line.
There is a negative cell line also, so if you compare, that will be very interesting.
HPV negative.
Yeah.
Yes, C33A, that we can do, C33A.
And I've got next question, that since you are finding so much interesting data of mitochondria, so if you got cell line which is a rho positive, like rho 0 cells, mitochondrial like level you go down, and then you say whether your sensitivity of radiation changes. If you deplete the mitochondria in cells and say they see the effect, that will be something very interesting to know.
Yes,
what is happening.
That is a very interesting idea, but I didn't do it. But
actually, I'm sorry, a very nice interaction was taking place, but we are late by 1 hour.
Yes.
So we can continue our discussions during tea time. So thank you very much for all the speakers for giving a very nice overview and also presenting your work nicely. And thank you, Dr. Singh, for being with us. Thank you very much.
Thank you, chairpersons. Thank you, speakers, for the wonderful 45 p.m.
Good afternoon, everyone. As time is of constraint, I request everyone to kindly assemble in the hall so that we can proceed with the next session. I repeat, kindly assemble in the hall so that we can proceed. Good afternoon, everyone. Hope everyone had a refreshing tea break. Please kindly assemble in the hall. We are about to begin the next session. Everyone, I repeat, please kindly assemble in the hall so that we can move with the session. The next session is on tumor radiosensitization by targeted genes. I request Dr. Hans Kreze from University of Amsterdam, Netherlands, and Dr. Rakesh Kumar Singh, Head, Nanotechnology Department, AK University, Patna, to preside over the session. Sir, as time is of constraint, please take over the session and kindly control the time.
Okay, good afternoon, one and all. In this session, there are 5 speakers, and the first speaker is from Ketes B. Katie, the University of Missouri, USA. And his talk on the title, The Radiation Research and Green Nanotechnology in the Discovery of New Medical Modality, the Nano-Ayurvedic Medicine, Preclinical and Clinical Investigation. Very good topics in the modern trend. The Government of India is also giving very special importance on these types of research nowadays. Now I request the Professor Katis B. Getty to deliver the talk. Sir, please.
Yeah, first of all, I would like to thank the organizers for this opportunity, inviting me and my wife, who is among the audience. I would like to thank the chairpersons for your kind introduction. And I want to use the same rule used by several of my overseas colleagues, an unwritten rule that every 10,000 kilometers you travel, you get 5 minutes extra. And I traveled 17,000 kilometers, so about 7 minutes extra. Gentle reminder to our dear chairman colleagues. Okay, the topic as you see here, and after listening to numerous eloquent lectures for the last couple of days, I deviated a little bit, not a whole lot. I'm going to share several slides for those of you involved in mitigation of radiation effects to the human body. I think we have something new to offer, something outside the box. So I want all of you to take a look at it. We can discuss during or after the lecture. Before I begin the discussions, some disclosures. Almost every aspect of the work I'm presenting today has been licensed to corporations through our patents, but we do not own any part of the company, any percentage of the company, nor do we own any of the stocks in those companies. So I just wanted to make sure that we do research at the university very similar to how we would conduct ourselves if we have had a federal grant from the NIH or from the Department of Energy or from the Department of Defense. Overall plan today, as shown here, as I said, I'm going to deviate a little bit. I'm going to share with you these very interesting chemical analogs made from amino acids, which in our opinion will open up some new opportunities when it comes to mitigating radiation effects. There were several lectures yesterday, some lectures today. I'm going to show you how those chemical structures look like. And then I'm going to delve into innovative approaches for dual radiation therapy and immunomodulatory therapy. Why do we need this? So we're going to talk about that. Then we're going to discuss very serendipitously, when we were doing radiation research, we thought we were strictly confined to radiation research, but it turned out that what we have been doing has a direct relevance for Ayurvedic medicine, especially in understanding the mechanism and enhancing the efficacy of Ayurvedic medicine. And then we termed this new medical modality as nano Ayurvedic medicine, approved by the United States Patents Office as a new medical modality. So I'm going to be talking preclinical and clinical studies. Many of you know that my university houses the world's largest university-owned nuclear reactor. We are not the world's largest, but we are the world's largest when it comes to ownership by an university. These are the attributes of the university: 10 megawatt. We are going all the way up to 20 megawatt pretty soon. Work is in progress. And our approach has always been, over the last 3.5 decades, we perform experiments to develop therapeutic probes, diagnostic probes, as shown here, and then we test them in small animals, very easy, very cheap. Then we do a little bit more adventurous. We test them in human mimicking animals, which is very vital when it comes to clinical translation. Very often in biomedical sciences, we lose sight, or we often miss the objective. The objective is not to treat mice. The objective is to treat humans at the end of the day. So that's why we do these experiments in human mimicking animals, where the disease mimics human diseases, both in terms of the genetics and morphology and so forth. So through this, we have been very successful. 6 regulatory approved products. Many of these products, I'm sure there are experts in nuclear medicine, radiation oncology within the audience. If you're using Quadramat for bone cancer, if you're using Ceratec for brain imaging, or if you're using TheraSphere for treating liver tumors, guess what? Every single one of those products is a direct discovery from my department. A lot of you don't know. You think that they have come from other institutions. They have originated from my institution. Today I'm going to talk more about the intervention of nanotechnology in medicine, as well as intervention of nanotechnology in mitigating radiation damage to the human body. This is what I'm going to talk about. It's not an advertisement. I have written the name cardipeptide. That was recognized by the National Academy of Inventors. These are 3-dimensional molecules, as shown here. Provides a plethora of ligands, whether you're interested in thorium, interested in actinium, radium, or simply trying to get the radioactivity out of the kidney. These ligands are non-toxic. These are basically multi-dimensional amino acids, as shown here. I'm going to show on the next slide. As you can see here, this is a compound with 3 alanine groups, both the D and L forms, a compound with 3 glycine groups. This is a peptide with 3 arginine groups. You can count the number of sites that it can bind to the metal, and we have used them very effectively to get the radiation out of the liver. If there is an increased uptake of radiation in the liver, by either injecting IV or by giving oral, these compounds go to the liver. Why? These are fully characterized. We have patents and publications on that. These compounds, by the way, are very unique. Not many compounds, especially there was a lot of discussion about DTPA, which is considered to be a gold standard because we simply don't have anything else. But DTPA, as was pointed out nicely by previous speakers, it's very hydrophilic. DTPA will a very small percentage would ever go to the liver. If you have to rid radioactivity of some metals from the liver, DTPA is not effective because the clearance of DTPA is mostly through renal. So for you to pick up something that you want to remove from the liver, you need a chemical molecule that is lipophilic. It has to have a hepatobiliary uptake. DTPA doesn't do the job. This does that job very spectacularly because it's an amphiphile. It has both the hydrophilic moiety as well as the hydrophobic moiety. In other words, it has water-loving and water-hating characteristics built within the same molecule. And the molecule is very small, which is important. Doesn't cause systemic toxicity. We have done lots of work on this, shown clearly that if we load up pig's liver or dog's liver with lots of copper, which is toxic, and then IV inject this molecule or orally deliver this molecule, we have seen clearance of that copper through the renal system. In other words, that molecule goes to the liver, picks up unwanted toxic material, which is a metal. If you have radioactivity, thorium, or other radiometals, picks them up and then clear through the renal clearance. So this was a deviation I just wanted to show all of you. And now we're going to start another interesting out-of-the-box approach. It's very vital that we talk about cancer. Some of you have already talked about cancer in this meeting because cancer kills more lives compared to TB, AIDS, malaria combined. So this is a burden. This is a huge financial burden, takes away close to 1.5% of the gross domestic product globally. We spend a lot of money. Still we see deaths of patients, despite all the advances in immunotherapy, name it, radiation therapy, site-specific monoclonal antibodies, site-specific proteins, peptides, we still have a lot of work to do. This is a responsibility of not one group, not one institution, not one country. It is a collective effort. So we need to be cognizant about that. Now, radiation therapy has been discussed. Again, Dr. Nagaraj Huilgold, the country's expert in radiation biology, is here. If I said too much, it is preaching to the choir. I need to learn a lot from him. But radiation therapy does its job. But it also creates a debilitating condition to the body, to the human body. It's well established. Radiation therapy basically creates a condition where the pro-tumor signaling factor, which is the NF-kappa B, is always elevated. There are no exceptions. Every patient going through radiation therapy, if you were to draw the blood and send it to pathological labs and analyze NF-kappa B, IL-10, IL-12, M2 and M1 macrophages, you will see radiation therapy results in pro-tumor cascades of events. Enhances NF-kappa B, in turn results in elevation of IL-10, which is a pro-tumor cytokine, results in pro-tumor M2 macrophage. So how do we mitigate these effects? The intention of radiation therapy is to treat cancer. But at the same time, radiation therapy is resulting in these unwanted adverse effects. So we need to be thinking about that. And we can talk about it. There are ways we can solve this problem. And also radiation therapy leads to opportunistic infections. No wonder that people going through radiation therapy as well as chemotherapy, those patients are advised to be very careful. Don't get infected. Why? Body's immune system has been already compromised because of chemotherapy and radiation therapy. So this is an unmet clinical need. How do we address these adverse effects of chemo as well as radiation therapy? How do we enhance the life-saving of patients when we administer these therapeutic modalities? We show that through various different means. Lots of people have eloquently spoken about lutetium and other isotopes. We use a very old standard. Gold standard, you can't beat it. But gold alone will not do it. We convert gold into radioactive gold. I'm going to be talking a little bit about gold 198, ideal half-life, pretty good beta. It also has a decent gamma that we can do scintigraphic imaging to monitor patient response and all that. And we nanosize this, a very different approach. Why do we nanosize this? That way, we and others have shown the tumor uptake of gold nanoparticles exceeds any other pharmaceutical modality known to date. You're going to see some of those results. Nanoparticles are important in medicine because they enhance therapeutic payload. If it is gold, each nanoparticle contains thousands of atoms of gold. Each atom can hold on to diagnostic or therapeutic probe, which means I can amplify diagnostic or therapeutic payload that is simply not possible by conventional therapies or conventional diagnostics. How do we do that? We use holistic approaches. What we discovered, one way to produce gold nanoparticles is to use toxic electron reducing agents. People use sodium borohydride or hydrazine, very toxic. But we use agents that are readily available to us. These plant-based phytochemicals are powerful antioxidants, and they have lots of electrons. When we mix these agents with the gold precursor, we produce gold nanoparticles. A well-defined size. This sounds like an interesting laboratory experiment. Now, every single one of these gold nanoparticle production processes is an industrial process. They are actually used for treating human patients. Every single process has been licensed to pharma companies. And each nanoparticle, as it creates a coating of a specific phytochemical, these phytochemicals have predetermined affinity to tumor cell receptors, whether it is GRPS receptors, laminin receptors, somatostatin receptors. They do target, but you can't use them as they are because you need to enrich these nanoparticles. You create hundreds of signatures of these therapeutic probes on each nanoparticle and then use those particles for either diagnostic, imaging, or therapy. This brings us an interesting connection. this was not intentional. If you really look at the annals of medicine, for those of you who have come from overseas, there is a take-home message. The concept of medicine actually started in this part of the world, 5,000 plus years ago. Lots of us don't know that. It was started through Ayurvedic medicine. But Ayurvedic medicine is so unpopular, despite the fact that it was started 5,000 years ago. Less than 1% of the global population use Ayurvedic medicine. Why? Obviously, there are problems. We're going to see that. What if we make this Eastern medicine, traditional medicine, Ayurvedic medicine, as effective as what we call it as the allopathic or the Western medicine? Why is that important? It's based on probability. If you have more number of medical modalities, you enhance the chances of success. You enhance the life-saving protocols significantly. It's a probability theory. But most doctors right now, they don't co-prescribe any of these medications alongside the Western medicine. There are problems here. So we need to build this bridge. Just a quick example, just imagine the pandemic is really fresh in our minds. What if we had alternative modalities to treat COVID-19? We all depended on one modality coming from the English medicine. We put all eggs in the same basket. What is the net result? We saw millions of lives lost. And it continues. What if we make these modalities scientifically rigorous, reliable? What if we made these modalities where we can actually expect clinical outcomes on par with what we know of Western medicine? That's an important question, a tall expectation. World Health Organization tells us that global population, they don't want chemical-based medicine. This is a survey. Global population would love to have herbal medicine. It feels good. But medical doctors who are here, they don't understand, they don't believe that Ayurvedic medicine would work as well as it is supposed to. So we need to look at what is that. I'm going to take an iconic example of Ayurvedic medicine. This has been used, it's a gold metal-based medication. In this part of the world, they call it as Swarna Bhasma, Sanskrit name. It is still used for treating asthma, cancers, muscular weakness, heart conditions, and all that. But if you look at the right side, there are huge problems. Reproducibility of formulations. You can't produce this medication exactly the same way, in exactly the same chemical constitution every time. That leads to undefined doses, which means you can't rely on the mechanism. And also high kidney uptake. It causes gold toxicosis, patients die. So this is one of the problems. If you ask me why this medical modality, despite the fact that it was discovered 5,000 plus years ago, why it's not used? These are the problems. And how do we tackle those problems? I'm going to show some example here. Radiation research looks like it comes handy. What we are doing here, we are producing carrier-added gold-198. We produce this in my nuclear reactor every day. It is used for brachytherapy, therapy of glioblastoma and so forth. But when you do the carrier-added production of this, only 5% or maybe slightly less of gold atoms become radioactive. 95% of the atoms still are non-radioactive, but it's still used. It's cheap. If you wanted carrier-free, then you have to produce gold-199, where the platinum-198 is the target. That's very expensive. But this does a tremendous job. I'm going to show you the results of that. The process to produce here, we have shown we use an electron-rich molecule coming from mango skin. Mangiferin produces gold nanoparticles instantly, identified, characterized extensively. We also have a patent issued on that. Mangiferin, non-radioactive mangiferin gold nanoparticles, and the mangiferin radioactive gold nanoparticles. It is a mixture by default. You don't do this. When you irradiate gold and produce gold nanoparticles by the previous reaction I showed you, you would always end up with this mixture. We focused on the therapeutic efficacy of this radioactive gold as well as the fate of non-radioactive analog in terms of what the non-radioactive particles do to the tumor cells and tumor sites. We want to really address these issues. Radiation therapy generally leads to enhancement of NF-kappa B, enhancement of IL-6, enhancement of IL-10. All of them are pro-tumor cascades. Ideally, a cancer patient would like to have enhanced IL-12, enhanced TNF-alpha, and reduction of NF-kappa B, reduction of IL-10, reduction of IL-6. So can we achieve this? We can achieve this if you took the mixture of radioactive and non-radioactive gold nanoparticles. Quickly getting into what happens when you injected these radioactive nanoparticles into tumors. You can see over 85% of the injected dose goes to the tumor. Very minimal, if anything, goes to any other non-target organs. Most importantly, when you're comparing the kidney uptake to what is known of gold drugs, traditionally, there is a 0% kidney uptake, which means that we have solved an important problem that has haunted medical community, the traditional Ayurvedic medical community, for over 5,000 plus years. If you converted gold metal into gold nanoparticles, you will have no fear of kidney uptake. You have solved an important problem related to toxicity. And then we did therapeutic efficacy studies in a host of different cancers. I'm just showing one example here. And what you're seeing here, the comparison of radioactive gold nanoparticles with the non-radioactive analog. This is the non-radioactive analog. The non-radioactive stuff is as good as the radioactive analog. This is where we got intrigued. And then we started probing. Yeah, that's the one I'm talking about. And some examples here, we also used other phytochemicals, and the same thing, if you used a phytochemical from a tea, e.g. CG, same thing. Uptake in the tumor is huge. That translates into excellent therapeutic efficacy as shown here. Control group, treated group, you can see there's almost a 85, 95% tumor reduction, tumor burden reduction compared to the control group. And injected activity resides within the tumor, as shown with this beautiful scintigraphic imaging images from prostate tumors in dogs. And this is the EGCG is another example where gamma-arabic protein was encapsulated, the same thing, control group, therapeutic group, excellent therapeutic efficacy. Just want to run through quickly. When we don't just take a plant and mix with gold salt and carry our experiments. That's not the idea. We identify phytochemicals. Based on the phytochemicals, we set up these protocols to produce nanoparticles, fully characterize them, not going to go through them for a want of time, and then test their in vitro stability, their ability to remain as a nanoparticle in the body, and then go about. So here's going back to that mangiferin example. We produce these particles as shown here, characterize them fully, and then again, it's a redox reaction, very well behaved redox reaction. This is how the phytochemicals assemble on the nanoparticle body. We create the radioactive equivalent as I described to you some time ago, and I showed you this picture some time ago. Now, when we saw this therapeutic efficacy as great as the radioactive analog, then we started wondering what is going on. Then we did this cellular targeting. These particles are tumor specific as shown here. They target the laminin receptors, very beautiful images. When you block the laminin receptors as shown here, particles do not enter the tumor cells. That means that what you're seeing here is not accidental. They are entering into tumor cells because these particles are site specific. Mangiferin targets the laminin receptors, which are overexpressed in a host of tumors, from glioblastoma to stage 2 to stage 4 pancreatic cancer, breast cancer, colorectal cancer, prostate cancer, and so forth. More therapeutic efficacy data in prostate and in breast tumors. At this stage, we asked this question, this tumor targeting, does it immunomodulate? Yes. When you look at the NF-kappa B, untreated cells and treated cells, NF-kappa B goes down. So there is an anti-tumor immunomodulatory effect exerted by these gold nanoparticles. And again, they target the splenic macrophage. As you can see here, again, I'm switching from radioactive gold to non-radioactive gold. scintigraphic imaging and then gamma counting helps us to understand the biodistribution, very little if any into the kidney. The presence of a reasonably high concentration of gold nanoparticles in the spleen suggests that it is targeting the splenic macrophages, and we did co-culture studies to show that these particles in fact convert pro-tumor M2 macrophages into M1. Again, I'm not going to describe in detail. These co-culture studies are very conclusive, and then these particles also enhance anti-tumor IL-12, suppress pro-tumor IL-10, suppress pro-tumor NF-kappa B. That means that they are immunomodulatory. Every immunomodulatory biomarker known in tumor biology we have studied, and these particles respond beautifully. We're allowed to conduct human clinical trials. These are triple negative breast cancer patients. Imagine, they had already gone through radiation therapy, chemotherapy. They had no chance of survival. And here are pre-treatment tumor burden, 90 days post-treatment. The government asked us to use doxorubicin in conjunction with our immunomodulatory gold nanoparticle, which is perfectly fine. When we did that, you see about 15% reduction in tumor burden. You may be wondering, it's not a whole lot, but again, quality of life, their eating habits, their body weight, social habits all improved. But what happens when another set of patients were only given doxorubicin? As you see here, the comparison is very clear. Doxorubicin, 90 days down the treatment, the tumors doubled, tripled, or quadrupled, or in some cases went up by 5 times. You should be comparing this adjuvant therapy, doxorubicin, with our gold nanoparticles, with only the doxorubicin. When you do that, you will see that the tumor burden just with the doxorubicin is so huge, we lost a lot of patients there. None of the patients died in this clinical trial with adjuvant therapy with gold nanoparticles. So I'm going to borrow about 7 minutes, gentlemen. So just
1 minute more, sir.
I was the chairman, so they approved for 7 minutes. Yeah, I'll do my best. I just want to go through quickly another paradigm. Here what we are doing is another phytochemical of significant interest. This phytochemical has a glucose backbone. We're very excited about it because tumor cells metabolize glucose, but the phytochemical has a therapeutic cargo. That means that if we encapsulated this on gold nanoparticles, there's a good chance that because of the glucose, which is metabolized so effectively by tumor cells, we will send this therapeutic cargo directly and selectively to the tumor cells. That was the hypothesis. We did all the things. I just want to show you how many of these therapeutic probes can I put on one nanoparticle. This is not a hypothesis. This is based on strictly mathematics. We use well-established equations. Typically, we can put as many as 7.6 into 10 to the 11 molecules per nanoparticle. Imagine the amplification of therapeutic payload. Do we have exactly this number on each nanoparticle? Perhaps not. But it just tells you the huge therapeutic payload that can potentially be put on each nanoparticle. Full characterization was done. If you're wondering what is the toxicity, cisplatin kills 100% of normal cells. And these immunoboosting gold nanoparticles within error limits, they're kind of non-toxic. I'll come to that in a moment. Just want to show you more immunomodulatory effect. They target the macrophage, which is important. The slide is important. We compromise the immune system in animals by administering cyclophosphamide, which is a well-known agent that suppresses the immune system of the body. Then we administered our nanoparticles. As you can see here, 2 different types of nanoparticles. There's a regeneration of the immune system in all the animals. These examples are from the spleen and from the serum. For this conference, of course, this shows the huge advantage tumor patients can enjoy while treating or using this for tumor therapy. But most importantly, this elevation of IL-12 is also vital for many, many different reasons, for mitigating radiation damage in human patients or humans exposed to radiation. These agents can be used for those purposes too.
Professor Kati,
Okay,
please,
I'm
could you
almost
go
done.
please go to your summary slide,
Yeah,
because
yeah.
we're
Yeah, I'm doing
running
that.
behind schedule. I'm very sorry.
Yeah, I'm doing that. So the animal studies, we ran up to 232 days, stopped the treatment on day 10. You can see the prophylactic effects also, not just the therapeutic effect. I'm going to say, so basically what I try to show you, the mixture of radioactive and non-radioactive gold leads to dual radiation therapy with immunomodulatory effects, so that the adverse effects of radiation therapy can be completely taken care of. We have gotten this latest patents, both American and European patents. I just want to run to the concluding slides. I just want to go to the last one, relevant for this meeting. These nano-Ayurvedic medicines, which boost IL-12 and regenerate immune system, can be used as drugs in achieving life-saving antibiotic and antiviral effects, as well as in mitigating the effects caused by radiation damage. I think we have built the bridge. Hopefully soon doctors will have the luxury of co-prescribing these medications. These are approved by regulatory agencies. Lots of people have worked here. I want to thank all the international collaborators, people from my labs, and numerous funding agencies. It has taken 30 plus years. I'm glad that we finally have tools to give it to the doctors for saving patient lives. This slide is important. We work in the US. Ayurvedic medicine in the US is like mixing oil to water. It doesn't blend well. That's why lots of people called us when we started. They ignored us, and they also laughed at us. Oftentimes they engaged in interesting discussions, but finally now we win the game, because it is going to save human lives, no matter whether one resides in the US, Europe, India, China, makes no difference. That's where John F. Kennedy's statement, it doesn't matter if you're standing on the shorelines in Mumbai or Chennai, the rising tide doesn't care whether the boat belongs to me or any one of you. Every boat goes up. So a rising tide lifts all boats up. And I think radiation research is a panacea in many ways to elevate the effectiveness of Ayurvedic medicine, and also make Ayurvedic medicine as one of the most relevant medical modalities when it comes to saving patient lives. With that, any questions?
Thank you very much for sharing your vision. Undoubtedly there are questions. For the remainder of the session, I have to announce that we will not permit any questions during this session, but so if you're interested, you can address Professor Kati during the lunch break. Yeah. So we will now move to the next presentation by Dr. Malia, and she will talk about, Dr. Maila will talk about tumor microenvironment targeting radiopharmaceuticals, a significant change from conventional approaches for management of cancer. Please come to the stage.
And his brief profile is on the screen. And Dr. Mala is he's from research domain is the radiopharmaceutical chemistry, the nuclear medicine, and organic synthesis. Sir, please.
Okay, thank you, Chair, and the organizers for giving me this opportunity. And I know that time is running short, so I will skip some of the slides, which I know most of you people may be aware. So, title of this talk you can see here, and most of you will agree with me that cancer management is going to be a 21st century challenge, and nuclear medicine, which all of you must have heard about, is going to play a vital role in this. This nuclear medicine is a medical specialty which involves the use of radioactive molecules. We call it radiopharmaceuticals, which can be used for organ function evaluation and diagnosis and therapy of various clinical conditions, including cancer. Of course, there are other diagnostic modalities, but the nuclear medicine has the advantage that it is highly a sensitive technique, and it can provide us with the functional and metabolic information that is happening inside our organs. Radioisotopes, this radio conference is all about radiation, so you know about radioisotopes. Radioisotope emit three major types of radiation, alpha, beta, and gamma. They have different penetrating properties, and based on that, gamma photons are used for diagnosis, and the beta and alpha particles are used for therapy. These are all known things. I am just skipping through it for the sake of completion. And this is a typical diagnostic procedure using radiopharmaceutical. You administer the radiopharmaceutical into patients, it distributes throughout the body, then it accumulates in target organ or tissues, and then we scan the patient to get the clinical information. But the question over here is, how can we make this radio tracer to accumulate in cancer or tissue? So you know different cancers have different receptors, and these receptors, and we can design molecules to target these receptors. For example, these two peptides, TOC and TATE, tyrosine 3 octreotide and tyrosine 3 octreotate, they can go and bind to somatostatin receptors, which are generally overexpressed in neuroendocrine tumors. So we synthesize these peptides, then radiolabel it with suitable radioisotopes, either for diagnosis or for therapy, and then we can use them for getting the clinical information, presence or absence of this cancer in the body, or even to treat them. But the problem with this receptor-based radiopharmaceuticals is that, as somebody previous speaker has mentioned, same type of cancer, different patients, and they have different levels of this receptor expression, which creates a lot of problems in terms of the efficacy. If they don't express the receptors, we cannot use that radiopharmaceutical for therapy. That is the whole problem. So this is one example where they have demonstrated that out of 27 out of 38 patient studies, they have shown somatostatin receptor expression, but out of remaining 11, in some patients they don't have any somatostatin receptor, or in some patients there are minimal somatostatin receptors, which creates a big problem for using these radiopharmaceuticals for therapy. So it is in this context that tumor microenvironment helps us with a lot of things like hypoxia, galectin-3 expression, VEGF, and so a host of things which can be a potential target for developing radiopharmaceuticals. Now, though there are so many, and the best part, that is why we are so much pursuing with tumor microenvironment based radiopharmaceutical, is that there is no significant variation in tumor microenvironment from patient to patient. Though there are several things, I will just touch upon two things, that is, one is hypoxia and galectin-3. It is our own work which we carried out in Bioatomic Research Center. So hypoxia is a common feature of solid tumors, and you may be aware that in its tumor, as the cells are as they move far and far away from the main blood vessel, they turn hypoxic, and these hypoxic cells are resistant to radiotherapy as well as chemotherapy. This hypoxia is responsible for recurrence in cancer, and therefore, this hypoxia is a very important clinical problem. And we do have some radiopharmaceuticals, which I will show you in the later part of the slide, which we can image hypoxia within the tumor, and this information can be used for hypoxia-directed radiotherapy. Now the question that we asked ourselves is that, can we develop radiopharmaceuticals to targeted delivery of radiation to the hypoxic cells? It's not just imaging, targeted delivery of radiation dose. So for that, we decided to use nitroimidazole, which is long been used for, which has shown hypoxia specific accumulation in cells. Like for example, this nitroimidazole enter the cells by passive diffusion. Once they are inside the cell, they undergo nitroreductase mediated enzyme reduction. But these radical anions, which are formed by this reduction process, are quenched by oxygen in presence of well-oxygenated cell. We call it normoxic cells. But they further undergo, these radical anions undergo further metabolism, break down and accumulate in hypoxic cells in the absence of oxygen. So this is taken care, and this is taken advantage of to develop this radiopharmaceuticals called 18-fluorine-fluoromizonidazole, which is used for imaging hypoxia in clinic. So just now we heard about gold nanoparticles. So we have used gold nanoparticles to develop some vehicles to transport drugs and radiation to these hypoxic cells. So we basically started with the gold nanoparticles functionalized with a lot of carboxylic acid. We used lipoic acid for this purpose, and then we tagged nitroimidazoles to these gold nanoparticles, and the job of this gold nanoparticle is to impart this hypoxia selectivity. We also tagged some macromolecules like DOTA. These are chelators where we can put radioisotopes, so to make it a radio tracer. And this also provides a platform for adding drugs or fluorescent tags. So this gold nanoparticle, as indicated in the previous indicated by the previous speaker, it actually becomes a vehicle that can transport a lot of things to the tumor cell. So our assumption is that if such a gold nanoparticle is made, they will selectively undergo hypoxia-specific reduction in hypoxic cells, whereas such an accumulation is not possible in normoxic cells. So now we made this labeled this gold nanoparticles with lutetium, and then carried out some in vitro studies in CHO cells. We kept the cells under hypoxic and normoxic conditions, and these radio-labeled nanoparticles were incubated with these cells, and here you can see that under hypoxic condition there is steady accumulation of these gold nanoparticles in hypoxic cells. To test it further, we made another set of gold nanoparticles, but this time you can see that there is nitroimidazoles are not conjugated to these molecules. When they are radio-labeled and similar study was carried out, then we see that there is no hypoxia-specific accumulation in cells, which indeed shows the role of nitroimidazoles in targeting the hypoxic cells. Now, to get more convincing evidence, the same nanoparticles were now put into a fluorescent tag was attached, and we carried out similar in vitro experiments, and we could actually show that in presence of hypoxia, these nanoparticles are accumulating in these cells, whereas in normoxic cells there is no accumulation. So to conclude this part, the in vitro studies demonstrated the utility of this nanoplatform for targeting hypoxic cell, and we have to check whether the same platform can be used to deliver therapeutic dose of radiation to hypoxic cells. Of course, we carried out some in vivo studies as well. Due to the paucity of time, I have not included those results here, but to be very frank with you, the in vivo studies are not so good. There is very fast wash out of these nanoparticles from the in vivo system, so that is a work in progress. Now quickly I will show other work on galectin-based galectin targeting radiopharmaceutical, again for tumor microenvironment. So this galectin is a family of small molecule effectors that control various biological processes like apoptosis, angiogenesis, immune escape, etc. And there are at least 14 mammalian galectins known, and people have worked with galectin-1 and galectin-3 most because they are found in most of the cancer tumor microenvironment. So each galectin has a carbohydrate recognition domain which can be used for active targeting purposes. So we worked on citraspectin. This is obtained from orange peels. They contain galacto-uranic acid to the extent of nearly 75%. And we are interested in this because galacto-uranic acid binds to the carbohydrate recognition domain of galectin-3. So the conceptualized core shell nanoparticle has a stearic acid core and a citraspectin-based shell, and we have envisaged stearic acid core because that can hold hydrophobic drug as well. But in this particular study, which I am going to show you, we have not loaded the stearic acid core with any drug, but it can be done. So next, if you look at the citraspectin, there are no functional groups sufficient to make pH-sensitive drug releasing molecule. So we partially oxidized it with sodium periodate to introduce a dialdehyde link. And the next step, a part of this citraspectin dialdehyde is modified to have some amine groups in it so that we can tag radioactivity to it. And finally, we mixed 1:1 ratio of citraspectin dialdehyde and the amine functionalized citraspectin dialdehyde in 0.1 molar borax to get our nanoparticles. I will not go into the details of how we exactly made it, but ultimately we get a core shell nanoparticle having both aldehyde groups as well as amine groups. Now they have been thoroughly characterized with TEM, SEM, and zeta potential, etc. has been carried out. They are found to be very stable in solution. And then we moved on to the drug loading part. So we took this core shell nanoparticle which are having the aldehyde groups and then incubated with doxorubicin, and we got the doxorubicin loaded nanoparticles. You note here that its pH responsiveness is coming from the imine linkage, which is after the conjugation of the aldehyde group and the amine group of doxorubicin. And we have carried out the release studies and can be shown that with decrease in pH, there is a steady release of doxorubicin in the system. We have carried out the IC50 evaluation, and it can be shown that these core shell nanoparticles are much more effective than the doxorubicin as a drug alone. Now, for the radio labeling, we use the Bolton-Hunter reagent approach, where since we cannot directly radio label the core shell nanoparticles with iodine, first we iodinated the Bolton-Hunter reagent, and this then incubated with core shell gold nanoparticles. You must have already noted that we had introduced some amine groups in the molecules in that core shell nanoparticle, which is now utilized now to tag the Bolton-Hunter reagent. We can achieve more than 80% radio labeling by this route. And finally, I will show you the cell binding studies with 131 iodine labeled core shell nanoparticles, which has shown concentration dependent cell binding. And the most important part is that we have also carried out a cytotoxicity studies. You can notice we have independently studied DOX-loaded core shell nanoparticles, then iodine 131 labeled core shell nanoparticles, and finally the DOX-loaded 131 iodine labeled core shell nanoparticle. And the graph clearly shows, maybe I can use this. And this graph here clearly shows that DOX-loaded 131 iodine labeled nanoparticles are behaving much better than 131 iodine labeled and DOX-loaded nanoparticles separately. Of course, to conclude this part, we have made this, it has shown a release of doxorubicin in a pH dependent manner, and it has also shown that DOX-loaded 131 iodine labeled core shell nanoparticles are much more superior than DOX-loaded and iodinated nanoparticles separately. And now the study that we are carrying out is whether by increasing the activity or loading of iodine 131, whether we can use this platform for chemoradiotherapy. That is, there is doxorubicin, there is 131 iodine, can be put together for better use. So thank you so much for your patience. If you have any questions, thank you.
Okay, sir. Thank you. And no questions because you are running to our session late. And the next speaker is the Chandan Kumar from Bhabha Atomic Research Center, Mumbai. And his title of his talk is The Approach to Improve Therapeutic Efficacy, Difficulties, and Outlook for Radionuclide Therapy.
So, very good afternoon to all, and thank to the chair and all my colleagues. So, I don't want to take much time. So, my topics is approach to improve the therapeutic efficacy, difficulties and outlook for the radionuclide therapy. Just before starting this brief description, so I just want to give the radionuclide, just Dr. Madhav has given brief. These radionuclides are only two types. One is diagnostic and therapeutics. The diagnostic contains the emitting this gamma radiation, and the therapeutic it emits they mostly emit both gamma and beta, and as well as alpha also. So this diagnostic has two types of one for this is specific for the SPECT and some are specific with the PET radionuclides. So this if you study how these radionuclides have been efficacy for this therapy, so we have to consider this LET. So this beta has also considered the low LET radiation, but still it has a range range in the tissues up to 13 mm or 12 mm. So it has also therapeutic efficacy, and compared to this Auger electron and this alpha, they are both considered as a high LET radiation. So these two, all these three are used for this therapy. So I don't want to go for the radiopharmaceutical because already I explained what is the radiopharmaceutical. So I just want to give because all this radiopharmaceutical we are developing, this ultimate aim is to use for this therapy and use for the cancer patient. So if we see this cancer incidence in this human, particularly in the male, these are lung cancer is higher incidence followed by prostate, colorectal, and in this case of female, the breast has higher and followed by the lung, colorectal, cervical and thyroid. So to consider this all these cancer incidences, we have to develop some radiopharmaceutical, and this radiopharmaceutical should means they are our aim should to how to increase the therapeutic efficacy, because you see there are a lot of problem happening because of this radio resistant and prolapse of the cancer. So our approach to enhance the radionuclide therapy efficacy. So there are the various approaches. So one is the how to use the, suppose there are some radiopharmaceutical which is emitting different types of radiation. So considering the different types of radiation, whether this is alpha emitting radioisotope using or beta emitting radioisotope. So by using that we can also enhance the therapeutic efficacy, and by using that same different isotope of different energy also you can consider. And third is the different types of tumor type, and finally the combination therapy. So you can go through this emitted radiation. So these are the one of my preliminary experiment were done in long back. So in that we have selected two types of radiation. One is a beta emitting radioisotope. This is using iodine-131, and this gamma radiation, this is cobalt-60 gamma radiation. So in this both cases we have selected 0.4 grade radiation, 0.4 grade radiation from gamma radiation and 0.4 for this iodine-131, and then we have studied the different gene expression in this Raji cell, and we can see this in the case of Bach's expression in this beta and gamma both are we can say they are similar to expression, but when we see this in P21 gene expression, we have found that there is a very different expression profile if you are considered for 30 minutes and 4 hours in this case of beta and gamma. So there is a very different we can say expression profile if you are using the same amount, means same gray 0.4 gray in the different types of radioisotope. And in case of RAD51 expression, we have found this is also very different expression if you using the beta and gamma. Where you see this in beta radiation, iodine-131, their consistent expression was there RAD51, but it is lower than this gamma radiation, but only for after 30 minutes, before 30 minutes and after 30 minutes also come down to the basal level expression. So here we see this 0.4 gray gamma, in 30 minutes expression is increased, but later it decreased, but in the case of beta there is a continuous expression. So these are the some of the experiment we can differentiate this, what are the difference between the effect of gamma and beta radiation if there is equivalent amount of doses delivered to the cells. So another approach is the energy of isotopes. So here this is also our preliminary study where you use the different types of radioisotope. And this is called 177 lutetium DO-TMP and ED-TMP. So both are this beta emitting isotope. And third one we have selected 188 rhenium HCDP. This is also beta isotope, but here this 177 lutetium is energy is around 0.5 MeV, but in 188 rhenium it is around 2.2 MeV radiation. So we have compared this therapeutic efficacy in the bone carcinoma cell line, this MG63. And we have found from this experiment, this PARP cleavage, this apoptotic DNA fragmentation study we have found done, and in that we have found that this lutetium 177 lutetium DO-TMP or ED-TMP, because both are lutetium isotope only. So their enrichment factor is around two times. But when you are using this 188 rhenium ED-TMP, it goes up to 7. So from that we can see this the higher energy of this isotope has more potential for the therapeutic efficacy. And similarly we have also studied this same cell line, different types of the cell cycle arrest, and in that case also found the lutetium is also doing this G2 arrest, but compared to this rhenium HCDP is very quite more effective because it arrests G2M more as compared to lutetium DOTMP. So from this one experiment, we can see this higher energy. If you are using any isotope for therapeutic efficacy, therapeutic purpose, so better to choose one higher energy isotope. But when you problem is there, this rhenium HEDP 188 rhenium is a can say generator based isotope, and it is very costly compared to this 177 lutetium, which is easily available from BRC. So the third approach is the tumor type. So in this also we have this our study, we have used that different types of cell line, around 4 cell line, this A431, MCF7, U937 Raji cell line. And in this also we have done some ELISA based estimation of DNA fragmentation. We have found this beta radiation, which is from iodine 131, and this 0.4 gray of gamma radiation, both has same amount of when the 0.4 gray of this beta and 0.4 gray of gamma. But in this both the cases we have found this iodine, this beta emitted from the iodine is more effective than this compared to this gamma. And in this all 4 different cell line, there is a differential DNA fragmentation also found. And even this PARP cleavage, we have also found there are variation in the types of cells. So in this case, we have found this Raji is this is a Burkitt lymphoma cell line. It is more sensitive, and that way is showing the better PARP cleavage, this enrichment, and even the DNA fragmentation also. So this is we come to the fourth approaches. We are using combination therapy. So in that we have selected this 2 or 3 different drugs. So I just want to give some glimpse of what I have carried out in our institution. So we have taken this one of the drug, this is doxorubicin, the well-known drug, and I have selected one rituximab. This is a anti-CD20 antibody. This is FDA approved in around 2000, in year 2002 or 2003, and we have labeled this rituximab with iodine 131, which is very easily available and is very easily labeled this any proteins. And we have studied this PARP cleavage, DNA fragmentation of most of the protein assay also, apoptotic protein assay, and the MAP kinase pathway also, and this Raji cell. And we see this in a doxorubicin, we have used 0.51 or 2 microgram per ml in this Raji cell, and rituximab I have used the 5 microgram per ml, and we have labeled this iodine 131 rituximab. It is around 3.7 megabecquerel per ml. So we have taken the equivalent amount, and we have found that when we are using this blend, doxorubicin is enrichment factor to up to 3, and when the rituximab was using, it was around 1.5, but when we are combined this iodine rituximab along with doxorubicin, so it is enrichment factor of the DNA damage, it will increase to up to 8. So it is quite means we can convincing you that when we are combined this our radiopharmaceutical along with these drugs, their DNA damage increases, and that may be more effective for the killing of the tumor cell. And likewise, we have studied this BCL cell expression and PARP cleavage, and again we have found that very good amount of we can say the expression down regulation of BCL and PARP cleavage were recorded. And we have also studied this some of experiment, this MAP kinase phosphorylation, and they found a very good down regulation of this phospho P44 and 42 in case of this combination therapy. And even this P38 also, they are found there is an increase of phosphorylation. So there are these are differential expression we have found when we are combined with this doxorubicin and this iodine 131 rituximab. Likewise, we also carried out with camptothecin and iodine 131 rituximab, where we also use this 250 nanomolar of this CPT, where it is around IC25 concentration. And in that cases, we have found this both if you are considering rituximab 1 or 10 microgram per ml, in both the cases, when you combined with a 3.7 megabecquerel of iodine 131 rituximab, we have find that a good amount of enrichment we attend at around more than 4 factor. And we have also done the cell cycle analysis, and in the cell cycle analysis we have found this the rituximab, it is causing the G1 arrest, and of course this when you combine it also quite more promising because it arresting the G1 arrest. And again we have done this BCL and this this phosphorylation of P38, and we have found that the downregulation of BCL-XL in the combination therapy, and there is this expression of this phosphokinase phosphorylation we have found in the combination therapy. So we have considered all these 4 factors how to enhance the cell death efficacy. So you can say this if you are considering the types of emitted radiation, so there is beta is more toxic than the gamma of equivalent dose, and even you can use this alpha. Alpha is also quite more toxic than this beta also. So and then next was the energy of isotope. So we have see the same dose of different energy has different effect, and this tumor sensitivity depends on the tumor type, because we are using different types of isotope, and this isotope has different sensitivity for the tumor. And when you are combining this radiopharmaceutical along with your chemotherapy, then there is a enhanced tumor response. So these are the first part, and second part we are going to see what are the bystander view of radionuclide therapy. So if you see this global market outlook of the nuclear medicine market, so this they are projected from year 2023 to 2024, this global nuclear medicine market is increasing from $8.94 billion to $10.19 billion US billion dollar. And this US is also projected in this by year 2032, this market has increased from 21 to 21.85 billion dollar. But there is also projected by the end of the year 2032, this global market of nuclear medicine will be increased from 8.94 to around 42.03 billion dollar. So there is a constant increase of this global market of the nuclear medicine, and this if you can see this market capture, so there's most of the global market of this nuclear medicine captured by the diagnostic radiopharmaceutical. So very few percentage you can see these are for diagnostic therapeutic. And even this nuclear imaging instrument also we have projected there will increase from 3.7 US billion dollar from year 2021 to 2026, it become 4.9 billion dollar. So considering all this, and when we see this total nuclear medicine clinical trial, so most of the clinical trial up to 2023 have found there are around 401 clinical trial going on this nuclear medicine, and out of there 146 are in the phase 3. But if you see this global use of the radiopharmaceuticals, so till date around 55 FDA approved radiopharmaceuticals are available, and out of that hardly 9 radiopharmaceuticals are used for the therapy, and some are for bone pain palliation. And if you consider out of that 9 radiopharmaceuticals, there are only 2 radioimmunotherapeutic agents has got FDA approved, and that is a long back in year 2002 and 2003. And out of that 2, one is out of market, so only one RIT agent, that is Javelin, is available in the market. So you can see this last 22 years, not a single RIT agent has got FDA approved, despite there are many FDA approved antibody are getting coming into the market every year. So what are the difficulties of this radionuclide therapy?
Can you wrap up, please?
Yeah. So there is a huge gap of this knowledge exists. That is why there is a long, there is a problem. We are not getting this FDA approved. So there's a first one is there you can just, because there is a lot of less knowledge about the energy, the bystander effect, abscopal effect are there, which is not, people are not working on that. Even there are also there are a lot of problem. There people are there is, you can say there are few paper available for different what are the different cytokines produced under this regimen, and there are there is a less study, or even can say there is a negligible study on this using this radiopharmaceutical. What are the cytotoxin, the radioresistance are there, and even this in case of the radio pharmaceutical or nuclear medicine, they are giving pre-dosing before the therapy. So pre-dosing and then followed by the therapy is just like pharmacists were studying this radiobiology. We are giving first lower dose and then given higher dose. There may be chances of radio resistance of the tumor. So this type of studies are not conducting in this nuclear medicine or radiobiology. So these are the lot of these gaps in this knowledge. So I just want to tell all these people of radiobiologists, please join hand with these radiopharmaceutical people and start working on this beta emitting radioisotope or alpha emitting radioisotope and their related radiobiological study, so that this in future whatever the clinical trial is going on, that will not fail from the market. So these are the despite the increased global market, demand of nuclear medicine and FDA approved R&D agent are limited. So because this most of the radiopharmaceutical are out from this phase 1 and phase 2 clinical trial, particularly in the case of therapy. So because there is a lack of radiobiological data, and so there is a need of radiobiological aspects of the radiopharmaceutical. So there is a huge gap of the knowledge. So there is required of lot of study regarding these radiobiological aspects. So if you are working on this radiobiological aspect of the radiopharmaceutical, it will be boosting the clinical data and there will be less chances of this failure of clinical trial. Thank you very much.
Thank you, Dr. Chandan Kumar, for completing your talk on time. There is no question due to delay in session. Now the next speaker is from Manikandan Selvaraj from General Datum Product Design Private Limited, Hyderabad. It is his technical presentation on the automatic alpha particle irradiator Bio Alpha Supreme. Yeah. And after that, the lunch break.
Thank you for the organizers. Actually, I am here to represent my company, General Datum Product Design Private Limited, Hyderabad. General Datum is a engineering product design company with a team of 70 plus mechanical engineers and also electrical, electronics and other department engineers also. Our expertise in electromechanical design, PCB design, embedded systems, and we are working on telecom and structural analysis, thermal and CFD analysis also. We are serving for our customers all over the world, especially in USA, Canada, Australia, and also we are serving for defense public sectors in India. Actually, we are part of a Janus 1 team. Okay, we designed and developed a 6U satellite which is in orbit, and also currently we are developing a CubeSat deployers, and this for the defense challenge IDEX, we developed this product and we presented this idea to IDEX also, and currently we are developing this product and soon it's going to be launched in space. So if anyone is working on any space biological research, we can help you in making it as a payload. And in our defense domain, we already worked with an American aircraft company in designing and developing rubberized fuel tank. And also for Bharat Electronics Limited, we are supplying composites materials. In composites, based on the requirements, based on what metamaterials they need, we can design, analyze, and we can make a structure or components out of it and give them. And this is all about our company. And with the BRC, we are working on this automated alpha particle irradiator, Bio Alpha Supreme. Actually, this we got a technology transfer from BRC. They have developed this technology, and we made it that particular product, product for a modern day usage. Okay, we made it very compact. We made it incubator compatible. so the researchers can use this product for various low dose and other radiation research purposes. Let me present our product details to you.
General Datum is a product design, engineering, software services, and research and development company. Our domain expertise is in defense, aerospace, power, data center, telecom, medical, industrial, automotive, and oil and gas. Our design expertise includes PCB design, simulation, valve design, IoT, embedded software, structural design and analysis, thermal and CFD analysis, satellite design, and composite design. General Datum has now collaborated with the Baba Atomic Research Center to engineer cutting-edge technology, the automated alpha particle irradiator, BioAlpha Supreme. BioAlpha Supreme is the first indigenous HMI-controlled alpha particle irradiator. Due to its innovative design, BioAlpha Supreme can achieve controlled, collimated, and uniform alpha irradiation. The system is designed to irradiate adherent mammalian cell cultures. However, in principle, it can be used to irradiate any material or samples, biological or non-biological, placed on a thin layer with a defined time and dose of alpha radiation. Now comes the big question: why BioAlpha Supreme? Understanding the health effects of alpha radiation is critical, especially in cases of accidental or occupational exposure. The use of charged particles, such as alpha particles, in targeted alpha therapy for cancer has drawn increased attention from researchers and clinicians aiming to improve cancer treatment with minimal side effects. Research on alpha radiation biology in areas such as space is still emerging and remains largely unexplored. Due to the limited range of alpha particles in air and water, conducting radiobiology experiments to irradiate cells under in vitro conditions presents challenges in achieving uniform and accurate dose delivery. Alpha irradiation requires specialized equipment, and while there are several alpha irradiators, including one at Baba Atomic Research Center, current systems face limitations. They allow only limited exposure time under ambient conditions and lack precise control over pH and temperature. Additionally, these systems are often bulky and heavy, making them difficult to handle and house. In response to these challenges, General Datum, using technology developed by Baba Atomic Research Center, is undertaking the design and fabrication of the BioAlpha Supreme, a compact, automated alpha particle irradiator. Unlike its predecessor, which measured approximately 1 meter and weighed around 50 kilograms, the BioAlpha Supreme is significantly smaller at roughly 33 centimeters by 30 centimeters by 15 centimeters and weighs just about 10 kilograms. BioAlpha Supreme's compact design ensures that it integrates smoothly with anaerobic chambers, laboratory incubators, and CO2 incubators. Cells can be irradiated under cultured conditions for prolonged periods, maintained at physiological conditions. Effortlessly control the irradiator with our user-friendly wired remote HMI feature, providing the user with precise and easy operation from a distance. Our flexible design allows users to use multiple sizes of specimen holders. 3 different specimens can be irradiated simultaneously using our 3-holder configuration, featuring an advanced interlock system that ensures flawless and safe execution of experiments, providing a fail-safe design. This cutting-edge device features a compact, lightweight, portable, and battery-operated design, making it effortless to carry to field experiments. Experience the convenience and efficiency as you explore new frontiers in radiation research. This technology holds promising potential across various scientific and medical domains. A few of them are researching radiation-induced bystander effects, exploring high LET radiation biology, investigating low-dose radiation biology, studying alpha particle-induced DNA damage response, enhancing plant mutation breeding, and exploring space radiation biology. Don't miss the opportunity to unlock the next level of innovation with our groundbreaking BioAlpha Supreme Irradiator. Elevate your research and achieve remarkable results today.
We have a stall over there. I request you people to come and visit our stall. We have a prototype of this model there, and we can do further discussions also on that product. And also, we can engineer your research projects into a products. So for that also, you can contact us. Thank you. Thank you all.
Yeah. And if any question can ask from you about product, any one question can ask about his product. Yeah.
Hi, Dr. Silvaraj. So that was a nice presentation. My question with regard to, can you please briefly describe your experience in CFD, which is the computational fluid dynamics, and how that can be translated to microfluidics in order to organize a payload for space work?
Okay. See, particularly on CFD, we are using an open form. Okay, we have a 10 members of team in our company who specially working on this CFD. Okay, so we are working on this thermal side actually. For thermal analysis, we are using CFD currently. Okay, so based on your requirements, we can further discuss with our team actually. Yeah.
Okay, thank you.
Thank you.
The delegates can visit the stall behind the conference hall, yeah?
Yeah,
okay.
yes. Thank you.
Thank you all the speaker for the nice presentation and talk in various topic related to this theme. Now, you are thankful from our chair for their valuable contribution and the nice presentation.
Thank you everyone. We will now break for lunch of 45 minutes and please be assembled at 3 p.m. so that we can resume the session promptly. The lunch is being served at the right side exit of the auditorium. Thank you.